Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome to the second day of International Conference, Empowering and Promoting of Underutilized Crops through Research Activity, Agro-Industry, Community Involvement, and also Digital Technology. On Thursday, 28th of October 2021, held by Underutilized Crop Research Center, UCRC, Faculty of Agriculture, University of Brawijaya. Praise upon Allah Subhanahu wa Ta'ala, the God Almighty, because of His blessing and grace, we can join today's seminar in a good and healthy conditions. And my name is Islamic. I'm the Master of Ceremony for today's conference. I would like to welcome to our Honorable, the Dean of Faculty of Agriculture, University of Brawijaya, Dr. Insinyur Damanhuri MS, the Vice Dean of Academic Affairs, Dr. Sujarwo SPMP, the Vice Dean of Student Affairs, Dr. Hagustano SPMP, the Chairperson of UCRC, Professor Dr. Insinyur Kuswanto MS, the Chairman of the Committee, Dr. Darmawan Saptadi SPMP, our Honorable Invited Speaker for the second day, Mr. Muhammad Fahmi Yunus, Ph.D. from International Islamic University, Malaysia, Professor Sim Sie from Guizhou University, Dr. Yong Wang from Guizhou University also, Dr. Nuawe Christian from Nigerian Institute for Oil Palm Research, and Dr. Prakisomta from Kassersat University. Our honorable electoral staff of Faculty of Agriculture, University of Brawijaya, our beloved students and participants for today's international conference. In the second day, we will also learn from the best invited speakers globally to frame promoting underutilized crops to research activity, agro-industry, community involvement, and also digital technology, so that this international conference will become a valuable sources for innovative ideas, concept, and breakthrough in developing underutilized crops in the future, especially in Indonesia. Excellencies, participants, there are several agendas for today's conference as follow. The first one, we will go directly to the invited speaker presentations, and there will be 2 sessions in these presentations, and it will be led by our moderators today. For the first sessions, our moderator is Dr. Darmawan Saptadi SPMP, and we have Dr. Afifuddin Latif Ajuriji SPM SC for the second sessions. And then for the next agenda, we will have the announcement of the best speakers, like what I said previously yesterday. And then we come to the closing statement, because this is the end of our international conference today. And then before stepping up to the main agenda, let's pray together first for the successful running of today's conference. So I will lead the prayer by saying, pray begins. Finish. Thank you, everyone. Now, before we go to the presentation sessions, I would like to invite all the participants to turn on the camera, because we will take a documentation. So I will need help from Mr. Adit to capture the screen. So are you ready? Okay, so I will count to 3. 1, 2, 3. Okay, once again, please give us your best smile, everyone. 1, 2, 3. Okay, thank you. Okay, so we will go directly to the main agenda today, the invited speaker presentations. Like what I said to you previously, that in this today's conference, we will have 2 sessions, and we will start the first sessions directly. And this session will be led by our moderator today, Dr. Darmawan Saptadi SPMP, for the first sessions. Dr. Darmawan, time is yours.
Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Very good morning, everybody. Warm greeting from me, Darmawan Saptadi, as moderator today. To all of participants, especially to the distinguished invited speakers. Today is the second day of the International Conference of Underutilized Crops. And yesterday we have listened and discussed many interesting things related to underutilized crops. Today, we will continue to listen and discuss other things, but still on the topic of underutilized crop. In this first session, we will have 3 speakers come from Malaysia and China. And before we start to listen the presentation, I will say hello first to all of 3 speakers. The first speaker is Mr. Fahmi Yunus, Ph.D. Are you here, Mr. Yunus? Mr. Yunus?
Assalamualaikum. Yes, I am.
Okay, thank you. How are you today?
I'm fine.
Okay.
How about you?
Fine, good. Yeah.
Okay, Alhamdulillah.
Okay. Okay, and then the second speaker is Professor Sinsi. Professor Sinsi, are you good today? Are you here? Not yet? Okay. And then the last speaker from this session is Professor Yong Wang. Professor Yong Wang, are you here? Not yet? Okay. It's okay. While waiting for Prof. Sinsi and Prof. Yong Wang, maybe they are still in process to join this Zoom. Let us start this session. In this morning, in about next 2 hours, we will be listening to this presentation of 3 speakers. So each speaker has about 40 minutes to deliver the presentation. And then we will follow by a question and answer session for about 20 minutes. We will start from the first speaker, but before Mr. Yunus start presentation, let me read his short bio. Mas Adit, please share the short bio of Mr. Yunus. Okay, Muhamad Fahmi Yunus, PhD, is the assistant professor of Department of Plant Science, Kuliah of Science, International Islamic University in Malaysia. And his area specialization is in plant biotechnology, plant tissue culture, agricultural science and technology, crop production and agricultural information system and technology, crop manipulation or genetic engineering, and then also transgenic plant, including transgenic crops from the aspect of pest and disease resistance, increased shelf life, color and nutritional enhancement, drought, salinity, and flooding resistance, and also mutation reading. Today, Mr. Yunus will talk about exploring the potential of Etlingera eliator, or torchinger, or in Indonesia we know as kecombrang or honje, as an ornamental food and medicinal plants. Okay, for not bothering Mr. Yunus' time, please, Mr. Yunus, the 40 minutes next is yours. Please, Mr. Yunus.
Okay, Assalamualaikum everyone. Good morning. Good morning and selamat pagi. So it is an honor to be part of this conference, and thank you very much to Dr. Dermawan for the invitation. So can you see my slides clearly?
Yes, yes, of course.
Okay, thank you. Okay, so inshallah I will start my presentation. Okay, and today's my speech is about the exploring the potential of Etlingera eliator, torch ginger as ornamental, medicinal plant and food products. Okay, let me introduce myself. Okay, I am Dr. Fahmi bin Yunus. I am from the International Islamic University of Malaysia, from the states of Pahang, Malaysia. Okay. So this is my outlines of my presentation. Okay, I divided it into 5 subsections. So I will start with introduction, followed by the economic potential, and then product development. Another 2 are the challenges and future perspective, and the last one for sure it is a conclusion part. Okay, so I will start with the introduction part. Okay, as you can see, so this one is a very beautiful flower. You can find it in Southeast Asia, in Malaysia, Indonesia, and also in Thailand. Okay, so this plant, Etlingera electio, or known as torch ginger, is a plant in the Zingibraceae family that is widely cultivated in Southeast Asia for ornamental purposes or consumed as food. Okay, so the name of the torch ginger comes from the striking resemblance of the inflorescence to a flaming torch. So this one, it looks really like a flaming torch or obor in bahasa Indonesia. And for this plant, they have different names. In Malaysia, we call it bunga kantan. In Indonesia, I think Indonesians call it bunga kecombrang. In Thailand, they call it kala. And for the Brazil, another tropical country in this world is Rosa de Porcelana. Okay, and it also can be found in Hawaii, USA, and also the Northern Territory of Australia. Okay, so it is from the Zingibraceae family. So you know, it is like curcum. This is like the ginger, turmeric. Okay, they are the same family with the ginger and turmeric. Okay, so as you might know, the Zingibraceae family is resilient to climate change with high adaptability towards different growing conditions. Okay, and majority of the Etlingera species is lack of ornamental value due to their short vast lifespan. Okay, they have a very short lifespan or the vast life, just for maybe for the 6 days for a blooming flower. Okay, and the torch ginger is one of the most known species of Etlingera. Okay, let me show you the close-up of the flowers or the inflorescence of this plant. Okay, so you can see here, okay, so this one is a true flower of the inflorescence. Okay, the true flower means that the stigma is located here. Okay, and the pink color actually is a bracts. So it divided between the outer bracts and also the inner bracts. Okay, so you can see that the true flower attracts different insects. Okay, to get this stigma from the plants. Okay, and the butterflies, Amygdalus bees, stingless bees, and sunbirds have all been spotted pollinating this species, and the pollen is located here at the yellow region of the true flower. Okay, so for this plant, it has a wide history of traditional uses in Southeast Asia, especially in Malaysia, Indonesia, and Thailand. In Malaysia and southern Thailand, the young inflorescence are used as herbs and food ingredients, and the extract from the stem is used to reduce swelling, while decoctions of the leaf are used to clean wounds. Postpartum women also use the leaf for bathing to remove body odor and wound cleaning. Okay, in Malaysia and Indonesia, the fruits are also used to treat earaches, diarrhea, cough, mouth sores, and also to increase the human appetite. Okay, and specifically in Indonesia, if I'm not mistaken, from the Sulawesi Island, okay, the pseudostorms were blended and filtered to get diluted and served to treat the typhoid fever patient. Okay, so but most important thing actually this plant is used for the culinary in Malaysia, Indonesia, and also in Thailand. Okay, especially the young inflorescence part. Okay, as you can see from this picture, it is used as a traditional salad in nasi kerabu Malaysia or ulam. Is it, if I'm not mistaken, it's similar to nasi ulam in Indonesia. Okay, so this one, the pink traditional salad. Okay, it is used in the nasi kerabu, and it also part of the ingredients in the sambal kecombrang in Bali in Indonesia. I think you can see here, I think the pink inflorescence. So it is part of the ingredients for the sambal kecombrang in Indonesia. And interestingly, in North Sumatra, if I'm not mistaken, in Medan city, the flower buds and the ripe seed pods containing tiny black seeds are used for cooking fish dishes. Okay, that's for culinary. Okay, and even though it is underutilized crop, actually it has a lot of economic potential. So for this part, I will share what can be done, what can we do to increase the potential of this underutilized crop. Okay. Okay, in Malaysia, okay, we have the Malaysia National Agri-Food Policy 2011 to 2020. Okay, this is the first NAP policy in Malaysia, and it's divided into 15 specific industries, and one of the industry is number 11, herbs and spices. And this plant is put under the herbs and spices to be developed into the industrial scale. Okay, the torch ginger is one of the 11 potential herbs to be commercialized, And interestingly, production of herbal plants in Malaysia for 2018 has reached a good value of 17 million USD. Okay, so this data I got it from the Department of Agriculture, Malaysia, in 2018 report. So you can see how importance of the torch ginger in Malaysia. Okay, you can see, you can make the comparison between the 2014 and 2018. For the production in metric tons, the 2014 data shows that it has like 905 metric tons in 2014, and you can see that there is steep increment to 1,787 in 2018, and it's same with the planting area. You can see that the number in 2014 is 178, and you can see the upward trend in 2018 when it reached the 356. Okay, you can see it's almost double when you compare it from 2014 to 2018. Okay, the average yield of this plant was reported at 6 metric ton per hectare with an overall production value of your 2.4 million US dollar. Okay, and the data I got it from the Department of Agriculture report in 2018. Okay, so we move to the next part, which is the ornamental, which is the the interesting part for this presentation. Okay, this plant has a lot of potential for ornamental value. I think, okay, from the picture you can see that it has a very bright color for this one to the pale pink color for this one. Okay, the varying shades of white, pink, and red colors of the bag and flower make this species an attractive ornamental plant. Okay, it is nice. I think if you can see it, it is a very nice flower. Okay, maybe you can use it during the Valentine's Day. Okay, maybe it is one of the potential that we can use. Okay, so I got this picture from my friend, which is Nur Asya Ismail, and he just published this paper in 2021. Okay, this one is the red, slightly red, and also the pink color, and it has a lot of variation for the flowers. Okay, this one is the plants, the Etlingera eliator, Bloody Mary. You can see it really looks like a blood color, and for this one is a gems race, and both of these cultivar is found in the northern territory of Australia, and I got this picture from the websites of the bamboo land. So this one you can see that a striking red color of the flowers, and they also like a mixture between white and pink. Okay, for this one, the Etlingera, different cultivar is a light pink, and this one is a Thai white, and both are from Thailand. So same, I got this picture from the bamboo land website, and interestingly, it also has a white flower of the inflorescence. It just shows that a different variation of the flower. Okay, you can see that this one is the white flower, the white bracts, and you can see this one. The yellow part is the true flower of this inflorescence, where the insects come to get the nectar from this part. Okay, so you can see like a different type of color and different type of structure, and it really has a big ornamental value. Okay, so development of new cultivars through breeding program. As you may know that if we have the flowers, I think we can try to cross it to produce a new cultivar. Okay, what happened to this one? Okay, in response to the floriculturist demand, extensive research being conducted, leading to more variation in the range of color, shapes, yield, and extending wildlife. Okay, this one is a different color of Etlingera eliator, and it mixed with the heliconia plant, so it is like a very interesting bouquet that we can use it to give a prize to another person. Okay, so attempt to create a hybrid was performed to produce its genetic variation, but it remains a challenging process. Okay, why? So I will explain later in the challenges part. Okay, so next part. Okay, if you want to do business, for sure we want to know about the yields, the end yields of the products, and some cultivars show a very good yields. Maybe you can use it for the production or mass production that we can sell it maybe online or in the market. So this ones that I gave like 5 different cultivars that have a very good yields, and they are Tropicas, Thai White, Ironstone, IAC 41, IAC Camry, and Yes, most of them are from Thailand, Northern Territory of Australia, and also from Brazil, if I'm not mistaken. So this one, the IAC 41 IAC Canberra. Okay, so that's for the year. So for me, it has a lot of potential that we can use it to sell this plant as an ornamental plants. Okay, in addition, okay, this plant has a medicinal values which has proven by the scientific report. Okay, you can search it on the journal and you can see that a lot of scientific reports about the medicinal values of torch ginger. Okay, for this plant, the flavonoids, terpenoids, saponins, and tannins are essential compounds successfully isolated from torch ginger inflorescence, the one that we use for culinary. Okay, and interestingly, extracted essential oil from leaf, roots, and rhizome of torch ginger also have been successfully isolated. Okay, for the anthocyanin, one of the flavonoid, okay, in the inflorescence can inhibit the ovarian cancer lines through cytotoxic activity. So this one is the pharmacological studies of the torch ginger. And plus, the ethanolic extract of torch ginger inflorescence has a vigorous antioxidant activity and could be used as a treatment for lead poisoning. And the last one, I think the flower extracts will also prove to repress human breast carcinoma tumor cell lines of MCF7 and MDA MB231 lines. So you can see that it has a potential to be one of the medicinal medicinal foods. So it is proven scientifically by the research. Okay, it has a lot of potential and it has been proved by the pharmacological activity study. Okay, so how about the nutritional properties? Okay, because we eat the inflorescence, so we expect that they have a lot of nutritional properties. Okay, so for the inflorescence part, the one that we eat, okay, it contains high level of protein, crude, lipid, and fiber, with the contents on dry weight basis is 12.6%, 80% also, 70% respectively, and plus in the inflorescence contains essential minerals like potassium, calcium, magnesium, phosphorus, iron, zinc, sodium, and also boron. So in terms of food products, I think this one is also a good suggestion to be one of the food for the future due to its high nutritional value in the inflorescence part. Okay. Go product development. Okay. Even though it is underutilized plants, but some researchers in Southeast Asia have developed different products, okay, for this plant. Okay, we will start with the agro-industry products. Okay, for in Malaysia, we have a small and medium enterprise, and they have developed several products for the food industries. Okay, as you can see here, this one is the torch ginger puree, or puree bunga kantan in Malay. And they also developed the sambal bunga kantan with ikan bilis, or the chili sauce mixed with torch ginger and anchovies. And the industry also has developed the dried torch ginger for cooking and also garnishing. Okay, yes, as you know that this one is important plant for the culinary in Malaysia. Okay, but for home essential, okay, they also have developed several different products, and we have in Malaysia the torch ginger essential oil, the torch ginger essential oil, and also soap, the bunga kantan soap or the torch ginger soap. Okay, so this one is the homemade products, the soap, and for this one is from the small and medium industry in Malaysia. Okay, so this one is the small and medium industry in Malaysia. But how about the big industry? Okay, interestingly, in Thailand, they have developed a skin whitening cream from the Etlingera eliator flower and leaf extracts. Okay, what most important thing, because you know that nowadays the cosmetic products are really popular around the world, across the globe. The same goes here, like everyone want to use the whitening products, the beautifying ourselves. Okay, so why don't we move to the big industry by using the Etlingera eliator flower and leaf extracts. Okay, this was I want to show the report from the Thailand. Okay, and they have successfully developed a skin whitening cream from this plant. Okay, why they choose this product? Okay, and why it is a good materials for the skin whitening products. Okay, sorry. Okay, for the flower and leaf extracts, okay, it has a natural antioxidant compounds along with having anti-aging, antimicrobial, anti-inflammatory, and tyrosinase inhibiting actions. Okay, these compounds, okay, in cosmetic products tends to be safer, biodegradable, and more environmental friendly. Yeah, for sure. They are not chemicals. They are mixed with chemicals, but use less usage of the chemicals because we use the natural products. Okay. Okay. Previous study by the same group shows that aqueous extracts consist of isoquercetin, catechin, and alkali acids, which are capable of inhibiting tyrosine activity, leading to a skin lightening effect useful in cosmetic applications. Okay, and it depends on the people. Whether some people love to look white, so maybe you can use this product. Okay, and they also publish the formulation of the FL1 cream from the Etlingera electro. So this one is their formulation that they use for their study. And they also have the dissatisfaction survey. Okay, you can see based on this survey that most of the volunteers are satisfied with the skin whitening product from the Etlingera electro. Okay, so maybe you can try these products one day. Okay, maybe. Okay, and another things. Okay, because we want to move forward to the big industry, so the researchers in Malaysia have tried to use it as a reducing agent. Okay, what is the reducing agent? Actually, it is used for the green synthesis of gold nanoparticles. Okay, as you might know, the nanoparticles is a new technology from the nanotechnology, and from this aspect is used for the medicinal purposes. Okay. Okay, what they have done, okay, the use of plant materials for the synthesis of gold nanoparticles, the AUNTs, okay, it is used for the reducing agents, used as a simple green synthesis of gold nanoparticles by using an aqueous extract of torch ginger. Okay, what does it mean by saying the reducing agents? Actually, as you might know, the gold ion, the AU, the gold ion is AU3+, and for reducing agents, they use it to reduce the ions from the AU3+ to the neutral form. So that's why it is called reducing agents. And for the gold nanoparticle, it is used in drug delivery, in biological and medical applications, plant genetic engineering, and also the sensory probes. And at the moment, the researchers in University of Malaysia from the same groups is currently developed the sensory probes in their lab. Okay, that's where I got the information from the personnel communications. Okay, so the another part of the torch ginger as underutilized crops is the challenges and future perspective. Okay, what is the biggest challenge for the breeding program for the torch ginger? Okay, due to the inflorescence structure, you can see that it is a big flower. Okay, the crossing process and its analysis is challenging to perform. Okay, the difficulty of crossing is proven with low fruit set due to the position of the individual flowers in inflorescence, identifying cross and pollinated flowers. Okay, more effective means of identifying individual flowers which has been pollinated is needed. So my suggestion is we can use the camera, the high-tech camera, to detect the individual flowers that have been pollinated. So we can see the growth and the development, and we can detect the seeds from the early stage after the breeding process. Okay, for this one is the future perspective. Okay, in Malaysia, the export value of Malaysia's floriculture industry stands at 119 million US dollar, which created 50,000 jobs opportunities. Moreover, Malaysia is ranked 8 in the world as an exporter of floricultural products. Thus, the expansion of the industry will increase national income and strengthen the national economy. But at the moment, most of the floriculture products in Malaysia is dominated by the orchid, rose, chrysanthemum, and also the carnation. So actually, it has a potential to be developed for the torch ginger, but it needs to compete with these most well-known ornamental flowers such as orchid and also rose. Okay, so Alhamdulillah, for the National Agro Policy 2021 and 2023, okay, the torch ginger is also part of the National Agro Policy. and it is still one of the plants that the government targeted to be developed into the industrial scale. Okay, inshallah, it will be launched by the end of this year for the NAP 2.0. Okay, this one just like I want to show the some picture of the farmers in Malaysia. So you can see that they have like their own farms, and they collected the inflorescence, the close inflorescence, to be sold in the market. Okay, and most of the products, most of the inflorescence sold in the market. Okay, so you can see some of the farmers have a new farm, so they just when they have the empty land, so they just put the toast ginger plants, and inshallah they will get money from it. Okay. Okay, what can we do? This one is the future perspective, or what can we do to improve the visibility of this toast ginger. Okay, germplasm collection. Okay, in Malaysia, toast ginger is mostly cultivated by small scale farmers with land-based varieties. And at the moment in Malaysia, we do not have, we have a germplasm collection, but it is not open for public. Compared to Northern Territory of Australia, I think they have a better germplasm collection, and also the data is available. Maybe we have to request to the Northern Territory government to get the data. And okay, that's think that for the Malaysia perspective, that's something that we can do it in Malaysia. Okay, the national germplasm and national database portal for collections need to be constructed to select traits that can be used in the future to generate more new cultivars. Okay, so after we have this germplasm collection in the state of Terengganu for the toast ginger. Okay, genetic improvement. Yeah, for sure. Okay, just like want to share some knowledge about the toast ginger. It is propagated asexually. It means that the plant is true to type. There has a similar genetic materials with the parents, because there is no mating between the female and male. So what the genetic contents is same, is true to type with their parents. Okay, to improve the genetic variation, okay, something that we can do is by using the tissue culture to induce the somatic variation, or we can do the genetic engineering where we insert the different genes from the plants into the parent plants, and also the in vitro mutation. In vitro mutation, such as we can use the gamma ray or chemical mutant agents such as colchicine, okay, to induce the variation for the flower, for the inflorescence, so we can get a lot more variation, and maybe we can do the selection of the flower. So yeah, sure, we need a more beautiful flower to be sold in the market. Okay. Okay, and modern households move forward to modern interior decoration. The successful inducing dwarfism of the ginger was the starting point to produce cultivar suitable for indoor plants. As you may know, during the lockdown, I think a lot of people stay at homes and they buy a lot of plants and they put it in their house. Because the toast ginger is very tall plants, I think if we can induce the dwarfism, we can get a shorter plants. I think it is better because we can move the plants from the outside into inside, and it can be one of the decoration in our house. Okay, so this one is the another thing that we can do is the in terms of farmer is accelerating modern technology applications. Okay, especially during the harvest period. Okay, optimizing farm size for productivity. Okay, based on my reading, I think there is no study about the optimizing the farm size for productivity. It is something that we can do in the future. Okay, establish an efficient agri value chain. Okay, this one depends on the industry, especially for the small and medium enterprise. Okay, strengthening specific industry for herbs and medicinal plants. Okay, this one I suggest about the food products, and also we can sell it as a supplement because it has a lot of potential. It contains a lot of biochemical compounds for these plants. Okay, enhancing value added in food industry and agro-related activities. Okay, and for the whitening cream or any other cosmetic product, what we can do is we can have a clinical test for toxicology assessment and mutagenicity analysis. Cosmetic for nutraceutical products. Okay, so for the conclusion, okay, for this plant, Okay, it is a plant with a lot of benefits. Yes, for sure, I have explained it before. And for the torch ginger, it can be planted into industrial scale. Okay, for this plant, it has lack of enemy. It is a very resilient plant. It can grow everywhere. Okay, and abundance of potential make this plant as a perfect candidate for the plant of the future. Okay, so just some publications. For the torch ginger. Okay, and the first publication of from me for this torch ginger is in 2012 and followed by 2013. And this year, Alhamdulillah, I have published the journal also in the torch ginger in AgriVita journal. At the moment, actually my postgraduate student is currently doing the correction for another journal of Etlingera of this plant, and inshallah it will be published early next year. Okay, so this one is the reference that I use for this presentation. And thank you. I would like to say thank you to my friends and team members. Okay, they are from the Malaysian Agricultural Research and Development Institute. My friend Nur Asya Ismail, and from my colleague Tamil Chelvam, and also the Zarina Zainuddin. They are both my colleagues, and my postgraduate student Nur Salsabila. Okay, you can contact me at fahmi.yunus@iaum.edu.my. Okay, thank you very much. I think that's all from me.
Okay, thank you very much, Mr. Yunus. It is very interesting topic, and I'm sure that our participants have more curiosity about this plan. The question and answer session will start after the presentation from third speaker, but participant can start answering a question by texting on the chat room, and Pak Musafan will record it. Okay, gentlemen, let's move to the second speaker, that is Professor Sinsi. I see that Professor Sinsi have attend here.
Everyone.
Yeah, Professor Sinsi.
Yeah, thank you.
Yeah, how are you, Professor Sinsi?
So I'm very fantastic. Good. Hello, everyone.
Okay.
Yeah.
Good to see you. Yeah.
Okay, I will share my screen.
Just a minute. We okay. Will introduce you first, and I will let you to present. Yeah.
Okay.
Mas Adit, please share the short view of Professor Sinsi. Okay. Our second presenter is Professor Sinsi. Professor Sinsi is a professor at the Department of Plant Protection, College of Agriculture, Guizhou University, Guiyang, China. And his research interest is in protein expression, purification, and crystal structure determination of plant filamentous pathogen effectors. And then also molecular mechanisms of interaction between plant pathogen proteins and small molecular chemicals or pesticides. And
Sorry, it's my time now.
okay, sorry for the lost contact, Professor Sinsi.
Okay.
Are you ready for present your presentation, please?
Okay.
You have 40 minutes to deliver your presentation.
Okay, thank you. Your introduction. I will share my screen now. So, hello everyone. My name is Xin Xie. I'm from the Guizhou University in Guizhou Province, China. I want to share. My title is about the sorghum. The sorghum bicolor, underutilized crop with high stress resistance for plant genetic breeding. So today I want to introduce my project from this 5 parts: background, research, in significant, and result, summary, and acknowledgement. First, the background. What's a sorghum?
Sorry, Prof.
Sorry.
We don't see your presentation yet.
So how about now?
Not yet.
Wow, I think it's a internet.
We just see the window overview.
I will check.
Maybe you can try to close it first and then try to reopening.
Okay. So how about now? Can you see my slide?
Yes, good. It's okay.
Okay.
Please continue. Yeah.
Okay. So I will my title is about sorghum bicolor, underutilized crop with high stress resistance for the plant genetic breeding. I will skip this and the introduction, the background. The background, the sorghum is usually used for wine making, bioenergy, and feed plant industries worldwide. And it's an important crop, and this cereal is now widely grown in Guizhou province in China, and mainly for a winemaking crop. And this is a planting area and yield in the Guizhou province from 2019 to 2022. We can see from this chart the planting area and the yield is increasing year by year. And but we face a lot of problems. The problem in the field is that this crop is facing by the disease, the insects, the drought, and the salt stress, and this stress will decrease the quality and the quantity of this important crop. We can see from this figure, the left one is the figure I took in last year in 3 districts, and in the field the sorghum was destroyed by the disease, and in the right one is the sorghum was bitten by a insect. So we face a lot of problem. And this picture we took in last year in Xifeng district in Guizhou province. We can see the farmers near get nothing because of the fungal disease. Now I only can say it's a fungal disease. I'm not sure which kind of fungal, but it's a disaster for the farmers. And this figure is also in the last year in Tongzi district in Guizhou province. We can see the insect, the armyworm, even eat everything, they bite everything. This is the early stage of this in last year. And we can see still have some leaves, but in the late stage, only the stem is in the field. The others, the remaining is gone. So it's a disaster. So we face a lot of problem. So my interest is first, I want to share today is about the molecular mechanism involved in sorghum resistance by evolutionary method. And the second, I want to understand the molecular mechanism of disease-related genes involved in sorghum resistance. And the last, I want to share with you we use a novel approaches to engineer durable resistant sorghum cultivars. And first, part one, the evolutionary mechanism of a plant PLC. I want to share one of my project of the PLC gene family. And before this project, we know the plant evolutionary path is very complex. From this figure, we revealed that the plant orange and the evolution path is kind of complex from for example, from the primary endosymbiosis into the embryo biofata and to the true food formation and the flora formation, and to different kinds of plant, for example, dinocots, the monocots, the monodies, and the AA branch. So during this process, it's very complex, and we reviewed that during the process of plant evolution, and a lot of genes are involved in this process. A lot of resistant genes, for example, during the chlorophyta, and the TLP genes and the MYO genes, CYC genes, it's a big gene family. And in this gene family includes a lot of genes. And some genes also related with plant hormones and stress. And during this process, the plant will trigger these genes to help plant to resist the biological stress, for example, the disease or the insect, the plant hormone stress, and the abiotic stress, for example, drought and heat, cold, and something, and the drought stress. So it's the plant resistant pathway, and the plant resistant net is very complex. From this figure, we can see it mainly includes 3 pathways. The left one, for example, one star genes we call the XA21. It's a big gene. It's a star gene, and it's a resistant gene in plant which can receive the signal from the pathogen and transfer this signal inside of to plant nucleus, and to trick the broader resistance. And in the middle way, we can see a bunch of genes are involved in ROS production. For example, rank 1, spin 6, SPL11. This kind of genes are related with the plant ROS product. We know that the ROS is like a trigger in the plant. If plant met or face the stress conditions, it can produce the ROS, and the ROS can trigger the innate immunity in plant. And also in this process, a lot of NLR proteins or this kind of resist proteins are involved in this process. For example, PI9 and the PI7, PI56, and also in the right one we can see also a lot of non-NLR proteins are also involved in this process. For example, PI21, LR34. All these from this review we can see that the process of the resistance pathway is very complex, and but I want to share an example about the CBL and the CIPK. So CBL and CIPK is two big gene family. The CBL is calcineurin B-like protein, and the CIPK is a CBL interacting protein kinase. We can see from this phylogenetic tree, and in different species, include the numbers of the CBL and the CIPK is different. For example, in the Silagina, and only include 3 CBL genes and 5 CIPK genes, but in the soybean, glycine include contain over 15 CIPK genes. So we want to know why the gene number is different in different species during the evolutionary. So the one reason may be caused by the whole genome duplication or the whole genome triplication. So, but still we don't know. And I want to share one example is our previously work or our resist work about the plant PLC gene family. PLC gene family is PLC is a phospholipase C, and this gene family can be different or diverse. We can see in this gene family, it can be divided in 2 groups. One group we call the NPC, and one group we call the PIPLC. The NPC means is include non-specific phospholipase C domain, and in the PIPLC, it includes the specific phospholipase C domain. And we found that we found that in the bacteria and the plant, it has a typical phospholipase C domain in ABC group, but in animal have nothing. And for the PIPLC, both in the bacterium, plant and animal, they contains the phospholipase C domain. But we can see the domain is diverse, it's different. Also, we can see from the red figure, the structure for the NPC and the PIPLC is much different. And so we also reviewed the PLC functions of the PLC genes. And we found the PLC genes is really complex in the signal transduction pathways. For example, the PLC can receive the signal from the outside triggered by the plant hormones, for example, the SA, the BTH, GA, ABA, and they can receive this signal and transfer this signal instead to the plant nucleus and some signal and may have interact with the protein, for example, the COIL1, GAZ1, and TIR1. And also they have a relationship with a raw signal and have a relationship with the defense response triggered by the pathogen. So in this review, and we found it's really complex. And this is our previous work, and we searched nearly over 62 plant genome, and we tried to get the whole complete PLC genes. And we find in the non-angiosperm in PLC protein from the algae, first mosses, and geosperms are very clearly separated. And we can see from the left figure, in the angiosperms, the PLC group can be divided into 9 groups. And for example, MPIPLC1, the M means the monocots. So it's kind of can be divided into 9 groups. And more, we want to learn the expansion mechanism or expansion way and plans of this PIPSA implant. And we find the from the left part, we can see the expansion plans of the PLC cannot be affected by the phytosynthetic type, but by the polypolydization. The PIPSA genes are trying to reduce or trying to lost something during the plant evolutionary plans. And also we compared the similarity between the protein different species of we find nearly 14% similarity of PIPSA genes in species. And it's not very high, but still we can find some conserved domains. And also we want to check whether these PIPSA genes can respond to implant against with stress treatment and stress response. And we checked from the big data from the NCBI database, and we truly find that a bunch of PIPSA genes can respond to the abiotic stress, for example, the cold, the darker, the drought, heat, the salt, UV, and wounding, and also a lot of proteins are response to the biotic stress, especially the disease invasion. And from the red panel, we can see the different color. The red darker in the means there are more genes expressing or active, and the white one means the genes is not active or not expressed. And we can summary from the red panel, and we can summary that a lot of genes, PIPSA genes are upregulated after the drought, salt treatment, or abiotic stress treatment, and biologic stress treatment. It gives me some hint that this kind of genes maybe can be used for the molecule breeding in the sorghum. And we want to know the evolutionary mechanism of these PIPSA genes, and we construct the big phylogenetic tree, and we can summarize that the complete PIPSA genes already exist in the early stage of the plant evolution, for example, in the red algae group. But during this process of evolutionary plants, the completely PIPSA genes was lost in the plant group, such as Dunarina and the double star algae. And this is our hypothesis. We just still now we only get this data. So we give our range hypothesis about the PIPSA evolution, and we summarize the number of the PIPSA genes, and the NPC genes is different in different groups, and during the plant under the stress treatment, and the plant retain fewer PIPSA genes, but have more NPC genes. They are opposite. Why? Still now we don't know. But the next step, we are trying to do the functional verification. Our next step is we will use the transgenic method, or we will use the CRISPR, for example, the CRISPR method to knock out the candidate of PIPSA genes, or we can overexpress some PIPSA genes in the sorghum, or in the rice, or in the tobacco to see the phenotype. So this is our next step, and actually we are doing this work, and we will it will take me nearly 2 years or 3 years to get the functional result. So this is my first part of the today talk, and the second part I want to share is about the another project about the molecular mechanism of disease-related genes in sorghum. And I want to give one of example is about a big gene family. A gene family is called 14-3 gene family in sorghum. From this review, we can see the 14-3 genes is a big star. These genes can respond to abiotic stress. It can involve in primary metabolism and growth and cell division and plant hormones. And also it can respond to light or biotic stress. And we found a good reference from published in 2011 published in the Plant Journal. They found that if silence one of the 14-3 genes, 14E, the rice plants can enhance the resistance to the schist blight. So it's a good hint for us. So this is our approach. We want to use this strategy, and we first clone some the candidate genes and try to test the expression level or profile, and then we will express the protein and purify the protein in the bacterium. In the last, we want to get the protein crystal or protein structure and get the interaction protein from the sorghum. And this is our recent work or previous work, and we found in the sorghum it totally include or contain 6 members of 14-3 genes, and we got some conserved motifs, and we also detect the expression level or profile of this sorghum 14-3 gene family. When sorghum treatment or treated by the salt and drought stress, for example, we use a salt and we use mannitol, we use a PEG 6000 to treat the sorghum seedlings and test the expression level of these 6 genes. Actually, we found that some of the genes are upregulated and significantly, and still we want to test whether this gene family can respond to biotic stress. Here we use these two chemical. One is flank 22, one is catin. These 2 chemical can mimic the disease. For example, the flank 22 is amino acid from the flagellum of bacterium, and the catin is from the fungi. So when we use these 2 chemicals to treat the sorghum seedling, and after we check the expression level of the 14-3 gene family, we found that interestingly, and some genes are responded very significantly, and this gives me the hint these genes could be used as the potential resources for the next step for the functional verification. And actually we cloned the 3 of the genes. For example, we cloned the 14C, 14D, and 14F. And we also expressed. Sorry, I'm not sure everyone can hear my president.
Yes.
Okay, thank you. I'm not sure. Okay, so we also did to check the protein expression, and we tried a lot of conditions. For example, we use different strains of bacterial strains, and we check the different temperature in induction temperatures, and we check the chemical induction of APTG, and we got the soluble expression, and this is and the next we also get the protein purification, and from this figure D, figure E, figure F, we can see our protein is very specific, and the protein is purified, and so we can use this purified protein to screen the protein crystal, and we searched we screened over 1,000 conditions of thousands of different buffers to get the good protein crystal, and we luckily we get some good protein crystal. For example, index 66, and in different buffers the protein crystal grew very well. And in the next we used this protein crystal, and we did the X-ray diffraction. And this is a previous work, and we can see from this picture A to C, these three proteins get good diffraction result. But still, we are trying because we want to get the fine protein structure, and we are trying resolving this protein structure by the synchrotron radiation light source. So we are trying, and this process will take a lot of time. So this is our the second project, and for the next step, we will do more work about the interaction. We want to check the interaction between the candidate 14-3 gene with the whether they can interact with the plant phytohormones. And because we found that in the previous work, some phytohormones can activate or can upregulate these three candidates. So we want to check them in the protein level, and we will use the two methods, one called the MST, one called the ATC. This method can check the physical binding between the protein and the chemical. For example, IAA, for example, SAA or SA or ABA, this kind of phytohormones. And in the next, we will use some approaches, for example, isotohybrid, for example, QIPs and GST pull down. We will use this method to check the interaction between the protein and the candidate. So this is also our next step. This is the second project. Still now, I'm still trying this, because during this time, because this step, the screen step will take a lot of time, and we move to the third part, and I want to share is our previous work about the new cultivars. The part 3 is a durable resistant sorghum cultivars. I want to share with our two cultivars. One called the Moliang Noor #1 and one called Moliang Noor #2. These two cultivars showed very good quality in the field. And this is a picture we took in Zunyi district in Guizhou province. And this picture is Moliang #2. And this picture was taken in Tongren district in Guizhou province. So we can see the sorghum on the plant grew very well. And we also get the national certification for this new cultivars. It takes our group nearly for 10 years. And we also take this is our previous data about from the 2010 to 2012. And we can see in the field, this our new cultivars get good quality, and the quantity compared with CK. The CK is a regular cultivar in our province, and we can see in 3 years, each year, the quantity is over than 10% increasement. It's a big step, and we are trying to do better now, and we are trying to get more new cultivars to make the more quantity increasement or the quality increasement. And for the next plan, because the traditional genetic breeding will take a long time in the field, because we have to hybrid year by year, and because of weather in the north place or in some places during the winter is very cold. We have to stop this work, and so it will take a long time. But from my aspect, and my major is molecular plant pathology, and I will trying to use the molecular method to narrow down or to speed up this process. For example, we will use and we are using the CRISPR or the overexpress method and transfer our candidate genes into the plant, and to narrow down or to speed up the breeding period. Actually, in the sorghum is very hard. It's very hard to do this work, and we are trying to do to transfer the candidate genes. For example, the PIPSC genes are measured, and the 14-3 genes are measured, and transfer this into the sorghum or into the rice or into the tobacco to see the phenotype, to see whether the quantity or the quality in the transgenic line is much more sensitive or much more resistance. So this is our next work. And for summary, the part 1 I showed about my previous work about the gene family evolutionary path. We identified over 200 complete PIPSC genes from over 62 plant genomes. And for the PIPLC group, they can be divided into 9 types. And we found a lot of PIPSC genes are involved in plant stress against the abiotic stress. For example, the cold, the heat, the drought, and the UV warming. And also they are all some of them are involved in biotic stress, for example, the pathogen or the insect. So there could be the potential resources for our next step. I will choose or select some of them to transfer them to the plant and to see the functional reaction. And we also give our hypothesis about the arranged hypothesis of a PIPLC evolution model. We give our model. We can guess it may that the plant grew on if grew under the stress conditions, the plant retain the fewer PIPLC genes, but they are trying to get more NPC genes. So our next work is try to reducing or to knock out some genes, for example, in sorghum or some genes in rice to see the phenotype of the transgenic line of the PIPLC genes. And my next summary about the 14-3-3 project. In this project, we identify over 6 members from the 14-3 genes in the sorghum plant, and we also checked the expression pattern of this gene family when sorghum be treated by the abiotic stress, for example, salt drought, and biotic stress, for example, the pampas. And we found that 3 of them, 14C, 14D, and 14F, these 3 genes are upregulated significantly during this treatment. So we cloned these 3 genes and we express them in the bacterium, and we successfully get the protein crystal. And we are by hand job method, and we also get some previous work by the X-ray diffraction. In the next step, we will trying more about this candidate interaction with the phytohormones, or we will get the fine protein structure in the future. And acknowledgements, I thank for my lab members from the Guizhou University, Yuan Pengfang. He presented yesterday, and Mei Qin and Junmei also she presented yesterday. And I also thank for my other students, Qiaoli, Qinjun, Zhiguang, Xiaodong, Huanglei, Zerui. Also, I have to thank for my collaborators from the Guizhou University, Professor Ming Jian, Professor Xiang Yang, and also the Dr. Xue Qing from Shanghai Jiaotong University, China, also Bao Xin from the Kanai University, He from the Clemson University. These 3 collaborators give me a lot of suggestion about the protein structure, and thanks for their help. And I have to thank for the funding agency from the National Natural Science Foundation of China, the Science Technology Foundation of Guizhou Province, and the Advanced Programs of Guizhou Province for returned overseas scholars. So I thanks again for the organization from the ICUC, and thanks you all, and thanks for your attention.
Okay, thank you, Professor Sinse.
Thank you.
It's very interesting topic. I think your study is an advantage studies in sorghum because it's an underutilized crops.
Yes.
And I'm sure that this will have very broad benefits because sorghum is now become an important crops, including in Indonesia.
Yes.
In Indonesia, sorghum is known as one of the food source in some part of my country.
Yeah.
And so sorghum is.
So if possible,
also,
we can collaborate, or we can
Yeah. Here, sorghum known as one species that resistant to drought.
Yeah.
So they can cultivate at the marginal land.
Yeah.
And now this plant is also prospective to be a raw material for bioethanol.
Yeah.
Okay, maybe there will discussion further for the Q&A session.
Okay.
Once again, thank you, Professor
Thank
Sensei.
you.
We will move to the third speaker.
Okay.
Ladies and gentlemen, let's move to the third speaker from this for this session. But before we start, I will confirm first to Professor Yong Wang. Are you here, Professor Yong Wang?
Oh, yeah, I'm here.
Okay, thank you.
Okay.
Yes, okay, because before you present your presentation, let me introduce you first. Mas Adit, please share the...
Okay. Now I can begin? Oh, yeah. Okay.
Just a minute.
Okay.
Okay, our last speaker is Dr. Yang Wang, Professor Yang Wang from Department of Plant Pathology, Guizhou University. He is a professor at the Department of Plant Pathology, Guizhou University. Guizhou, China. And his expertise is in myology, experience in pathology and molecular phylogenetics and identification of fungi. Okay, let's start to hear the presentation from Professor Yang Wang paper. Please, Professor Yang Wang.
Okay. Thank you for the introduction of the holders. I'm very glad to take part in that this international conference. And my name is Yong Wang, or in Chinese, it's Wang Yong. And if later you have any questions, and you also can call my English name, Felix. My speech name is Disease of the Underutilized Crops and in Guizhou Province, the China. And my speech, and including 5 parts, then the background and the main and in Guizhou.
Sorry, Prof. Yang Wang, do you try to share the screen?
What? Yeah.
Do you share the screen?
Yeah.
We don't see it yet. Yeah.
Yeah. Now it's okay.
Not yet.
Yeah. Oh yeah. Okay. I will open it. So that. No. Where's my PPT? Okay, I open it. Now it's okay. Oh, yeah.
Not yet.
Oh, yeah. Okay. Now.
Yeah. It's yeah. It's okay. Please continue.
I will restart it. I will start it again.
Okay.
Thanks for the introduction of the holders. And my speech name is the Disease of the Underutilized Crops and in Guizhou Province, China. My speech contains including the following the 5 parts. And in the beginning, because then some listeners is not familiar with our universities, I hope spend 1 or 2 minutes to introduce the Guizhou universities. And Guizhou University in China is an old university, and now it's next year we will have the 110 years anniversaries. Now the Guizhou universities and covers an areas of the 362.22 hectares. It mainly includes the 3 campuses. And now Guizhou universities have the 40 colleges, which including the many majors. It including the liberty and agriculture and engineering and the law and economic, just like that. Now, for Guizhou universities, we have nearly 60 or 600 or 60,000 the students, and we have the full-time the faculty staffs is nearly 4,000. And if you have time available, and we hope to invite any of them to visit my universities. And we know, and in the world, there are many different kinds of crops and in different countries. And for the crops, in the growth stage, it will attract and by different kinds of the pathogens. It including the fungi, the bacteria, and the virus, and also the metals. And we showed the several pictures, and including the rice disease, and the wheat disease, the mango disease, and the potato disease, and this is the chili disease, and the banana disease. It in tropical regions, on subtropical regions, or the cold regions. And according to the data of the United Nations Food and Agriculture Organizations, the annual losses caused by different kinds of the plant disease to the global economic exist to $220 million US. So for the agriculture, the industry, and plant to solve the disease problem is very important for the continuous development. And sometimes we will discover that several or even dozens of the disease can be found on a single crops. And in human histories, and we usually found the dangers of the farmings. And the following, we showed some of the pictures. It caused by the potato leaf blight, and in Ireland, and millions of people died in that the disaster, and millions of people and moved from the European and to American. So and many mycologists and the plant pathologists and hope to solve the disease problems. And the Guizhou province and in southwest of the China's, it is a transportation hub in southwest China and an important part of the Yangtze River economic belt. Not like the Indonesia, and in Guizhou province, the climate is very good, and also the temperature is not high, and in summer. And in Guizhou, the terrain in Guizhou is high, and in the west, and low in the east. It tilts from the middle to the east, and south. It is known as 8 mountains and 1 water and 1 field. It means in Guizhou province and more than 80% the areas is belong to the mountains, and just 10% is belong to the spring or rivers, and only 10% areas is suitable for agriculture. And the climate is belong to the subtropical, the month climate, and we hope in futures, and you can come here to for some of the visit or to start some of the collaboration. And this is a map of the Guizhou province. Although in Guizhou province the farming areas is very limited, but we don't have a very big agricultural industry like north of China or east of China. But we also have our own characters, and in some of the underutilized crops. I hope to introduce them. The first one, just now, then the doctor shares and give introduction about the sorghum's industry. And by 2022, and the sorghum, the planting areas, and in Guizhou province, will reach 4 million mu. And mu, you know, it's a Chinese unit, and 1 mu is equal to 666.7 square meters. And among them, including 1 million mu of the organic certification areas. And in Chinese histories and the sorghums and for food, for one kind of the food for people, especially in north of China or west of China. But in Guizhou, it mainly as brewing sorghums, because in our province we have an important international brand of the liquor. We call it the Multi. It's very famous, and for the sorghum and mainly provide for the materials to brewing the liquor. And the second one, we know it's the walnut. It is understood that since 2010, Guizhou province has the vigorously development the walnut industries, and for solve many present in Guizhou province the poverty problems. At present, the walnut forest areas of Guizhou province is reached to 3.79 million mus. It have a very high economic values. Now, in this year, our province established the Guizhou Walnut Industry Innovation Alliance. Now I served as vice chairman and mainly responsible for plant protection. And the second one, this is the local local crops. We call it is coix, and for people usually to eat the seed of the coix, and we can use the coix to make some of the porridges. Because it is nutritious, it is suitable for all ages, especially for some of the old people or some of the patients. As the main traditional production areas of the coix, and Guizhou ranks the first and in the planting areas, and mainly in the Xinyi city. It is in southwest of the Guizhou province. And the total output of the COEX and in Guizhou province have reached 288,000 tons. And the output values has reached 4.3 billion yuan per year. And you know, this is the bean crops. And in Guizhou province, we have different kinds of the bean crops. We have the coffee and also the broad, the beans, and we can make the bean to different kind of the dishes. For example, we can make it as tofu. It's a very delicious or special food, and in Guizhou province, and for the hot pan, like that. And when it comes, I will have opportunities to bring you to taste them. Although I don't have the accurate planting areas numbers of this industry, but I believe and the planting areas is more than 2 million mu, and in Guizhou province. And this is we call it the buckwheat, and people often like to eat the seed of the buckwheat, and we can make it as a powders and to make noodle just like that. And because it have the function of to reducing the lipid and to prevent some disease. By 2020, the planting areas of the buckwheat have reached 1 million mu. And after, besides the above main crops, we also have other the underutilized the crops, but the planting areas is not very large. The first one we call it is the millet, and it's also a traditional Chinese cereals, the crops. And this is the peanut, and in Guizhou, the peanut is very red. Not very. It's a little small. We also call it the peanut, the peanut, and we use it as a one dishes. And when you want to drink some liquids. And the last one is waxy the corn. This is the main underutilized crops and in Guizhou, because for my works is a plant protection. So I hope to introduce my research. So I hope to introduce the disease of the underutilized crops and in Guizhou province. This is my work, because meanwhile, I am the post scientist of the, we call it is cost crops and in Guizhou province, mainly for the mainly to investigate the occurrence of the different kind of the disease, and also for some works about the diseased crops like that. Now we begin these topics that, and in Guizhou province, we mainly found the three kinds of the sorghum the disease, and the first ones and the purple the spots disease. We found from in the meantime, we will find the different pathogens belong to which might cause this kind of the disease, and we can introduce them and later. And the second we call it is the smart disease. And previously it was very severe. Now we use some of the agriculture approach, and now the occurrence of this disease is lower and lower. And this is the leaf blight, the disease. And the picture and showed and my master students for the investigation of the disease occurrence and in the field. And the second one is the walnut disease. And from the pictures, you know, and the disease can attract the fruit and the leaf and the stem. And the fruit we caught is fruit rot, the disease. And the second is a leaf blight disease. And the third one is a dieback the disease. And also it have the leaf spot disease. It might caused by the fungus or sometimes also including the bacteria. And the third one is a coix, the disease. And the first one is coix, the leaf blight, the disease. It is very serious and for this industry. And the second is the smart disease. And the third one is a new disease, and we discovered 2 years ago. And in these 2 years, it become and more serious. We call it the thrush blight, the disease, and usually, and you can observe the sclerotium with a high humidity and in the field. And this is the millet. In millet disease, there's have the fewer disease, and mainly we call this downy meadow. In Chinese, we also call it as the white is the hair disease, just like the hairs. And the last one is the leaf spot of the millet disease. And this bean in Guizhou province, they have different kind of the beans, and commonly, and the disease is the rust the disease, and we can observe the pathogens in the backs of the leaves. And this is a spot. This is the leaf spot, the disease. It also has the downy, the meadow disease sometimes, and you can observe the heifer in the backs of the disease, and also some the stem, the blight disease. And besides the following the disease, the symptoms, and for the bug disease, and we usually have the leaf, just one kind of the leaf disease. And for the peanut, that's mainly including the three kind of the disease. And this are also we call it as the peanut sclerotium, the blight is the disease, and the two kinds of the leaf. The first one is the brown spot disease. This is two common disease. This is a Sorghum. And except besides some of the disease investigation, we also for some of the pathogens identification. This is one kind of the leaf spot of the sorghums, And we collecting some of the disease the samples and to isolate the pathogens and purified. And we observed the pathogens and from the microscopes, and we can observe that conidia and conidia forest, yes, that different kind of that we can see the conidias and with the transverse sept and also some of others the septors, and we can after the morphological observations, and we know it belongs to the Etlingera. For the Etlingera, it's an important the pathogenic genus which can cost hundreds of plant disease. And meanwhile, we also can for some of the sequencing works, and we use for the for ITS, the GPD, and also Etlingera ALTA1, the gene regions for their phylogenetic analysis. And from the phylogenetic trees, and we can make a conclusion that and the pathogen belong to the Etlingera agnatas. This is the first report and in Guizhou province. And this is the purpose, the disease we have. I have introduced and from the last the above the subsections, and because now in Guizhou province it is more serious and serious, and we use the high throughout the method to clarify their pathogens. From the result we know it includes the Cercosporus, the pathogen, but it only occupied a low the percentage. We also for the isolate and the purified, we don't got the Cercosporus pathogen. We only got the Paslotipsis and the Cladosporium Etlingera and Didymela and Apicom and the Fusarium. My student is do the works, and for that the critical task to clarify the real pathogens of this disease. And we also do the works about the rest the disease of the bean crops and in Guizhou province. We got the pathogens and from the broad the beans and also the beans, and we got the two different kind of the pathogens, and you know that have some the morphological difference, and we also find that the DNA sequencing about that. It belongs to the Euromyces, but belong to two different pathogens. And for the walnut, the fruit rot, the disease. In the beginning, we think it caused by the cladotrichomes, but after the morphological observation, and we know it in fact and belong to the Botryophyria, and we also use the morphological description and also the phylogenetic analysis, and we found, in fact, the pathogen is belong to the Botryophyrias, the Docidias. This pathogen is also very important, the pathogen which can cause the hundreds of the disease and in different the plant hosts. Yeah, I nearly finished the introduce of my speech, my research content. And finally, I hope to introduce my research group. It's including the different aspects of the plant pathogens, and they are very young. We have a very young research groups. I hope in futures, and if we can find some interesting topics, and we can start some collaborations about the plant protections. And now in our research groups, and we have the PhDs and from different universities in Guizhou, in China. Meanwhile, we also have some PhDs and with the overseas experience, and they focus on the fungi. It is mainly research directions, and in our research groups, it's including the plant, the pathogens, and also the fungal diversities. Just like the Dr. Xie, it focuses on the molecular biologies, and also some PhDs and focus on the biocontrol, and also the edible fungi. Now it's a very important the industry and in Guizhou province. And for the last part of acknowledgement, and now for my research teams, it's including some foreign postdoctors. Now I have 4. It's from the Pakistan, from the Sri Lanka, from the Egypt, and from the Egypt, yes. And we also have some of the PhDs, because now I do some collaborative works and with Mafelang University in Thailand, and we have some of the PhD candidates and from that universities, and I also have 1 PhDs and from the Pakistan, and also I have nearly 10 the master students. Because for our research group, especially for me, to begin some of the international works is very important. Now our research research teams, no, our labs is have the one base. It is the Guizhou Guizhou's plant pathologies, one belt, one road, international cooperation cooperatives base. Now we have begin our collaborative works, and with more than 10 universities, and we also have an international journals, and name is Plant Pathology Quarantines. Now, although it is not SCI journals, but I hope in the coming 3 or 4 years, it can become an important the plant pathologies journals in the world. It mainly report the pathogens, the identification, and also some other topic about plant pathologies, and also some the mycological report or maybe other works. We hope in futures and these journals can get some research contributions from the participants of this international conference. Besides, we also hope to say thanks and to some international, to some not international, some of the grants. It's within the NSFCs and from China, and also some the Guizhou's grants. Okay, I hope to thanks, and I also hope to invite you again to come to China, to come to the Guizhou's, and I hope the plant pathologists and in Guizhou University is opening the platforms, and we hope to have more friends and more colleagues, and for our futures the research works. Thanks again.
Okay, thank you. Thank you very much, Professor Wang Yong.
Yeah.
Thank you for your invitation.
Okay.
And I'm sure that your students have presented the part of the research in this conference yesterday. Okay, that's nice topic. And from this presentation, we get any information about the other kind of underutilized crops in terms of species or the related diseases. And also we get new understanding that the category of underutilized differ from one region to another. One species can be categorized as an underutilized crops in one region, but not in other region. Okay, once again, thank you very much, Prof. Wang Yong. And ladies and gentlemen, we have listened presentation from 3 speakers, and now it's time to move to the next session, that is discussion session. We still have about 35 minutes to discussion, and we found some question on the chat room. Maybe we can the speakers can give a response first to the question from the chat room. Please, Pak Musafan, you can. Okay. The first question is pointed to Dr. Yunus from Vina Mafazatul. What's the featured composition contained in the whitening cream product made from ginger, torch ginger, yeah, which is produced in Thailand, so that this can be claimed to be a safer and biodegradable product, and has the product been well received by the public and has become one of the leading cosmetic products, especially in Thailand. Please, Dr. Yunus, you can, oh, sorry, we can continue with the second question. And Dr. Yunus can give response in one event. The second question is from Miss Maya Melati. She has 2 questions. The first one is torch ginger a perennial plant, so the flower can be produced for years? And the second question is, when the harvest time for flower, for food and cosmetic purpose, when the flower is blooming or before? Is there any comparison of biochemical compound content between the 2 phases of flowering? Please, Dr. Yunus, you can give the response. Mr. Yunus?
Yes. Okay, if I'm not mistaken, I got 4 questions. Okay, so let me answer the first question. If I'm not mistaken, it is from Miss Vina Mafazatul Ula. Okay, in terms of safety, okay, actually the research claim that these products are safer, more biodegradable, and more environmental friendly compared to the another products. Why? Simply because most of the products in Thailand use totally like chemicals to produce their plants. But for their products, the whitening creams, they mix with the extract from the torch ginger. So that's why they claim that they are more safer compared to the 100% cosmetic products from the chemical compounds. Okay, if I'm not mistaken, they have the person have another question. So let me explain. Okay, actually we are from the Southeast Asia that we have like the brown skin, and this brown skin come from the pigment called melanin. Okay, and for the melanin in one of the enzymes that produce this kind of brown color for us is like tyrosinase. Okay, for the tyrosinase, okay, for the extract from the torch ginger, they have the tyrosinase inhibitor activity. And this inhibitor activity comes from their phenolic contents, which is isoquercetin, catechin, and also the gallic acid. And that's why it is suitable for the watering product. Okay, I would like to say it again that it is safer, more biodegradable, more environmental friendly compared to 100% products comes from the chemicals. Okay, and I think the Miss Vina Mafazatul Ula also has another question. Has the product been well received by the public? Okay, at the moment there is no report about the mass public atau more people about the quality of the products. Okay, there's no report about it, and whether it is has leading cosmetic products in Thailand. Okay, because torch ginger is underutilized crop, so it is not a leading cosmetic products in Thailand. It is still in early stage of research, so that's why they have to produce this product in 2019. And I think same like in Malaysia and Indonesia, they have different bureaucracy, different level of bureaucracy before the products can release to the public. Okay, and whether they have sold this product, I have no idea about that. Okay, and the second one is, if I'm not mistaken, is from Miss Maya Melati. Okay, thank you for the question. It is a good question. Okay, for torch ginger, it is a perennial plants. Okay, so the flowers are ready throughout the year, so you can harvest it when the inflorescence is ready. If you want more flower or more inflorescence, you can put flower fertilizer, so can induce more flower during that time. Because I am a plant tissue culturist. Okay, for the plant tissue culturist, the inflorescence is not a good sample during the rainy season, because end of the year usually Malaysia has a rainy season, and during the rainy season it will cause contamination if you use inflorescence as your sample. Okay, and Miss Maya Melati also has another question. It's about the different stages of inflorescence, if I'm not mistaken, the question. And for food and other purposes, okay, we use the young inflorescence or the close bud. Okay, young inflorescence, not the blooming stage. The blooming stage is the old stage, and at the end of the stage before they produce the fruits. Okay, okay, why? Okay, for the culinary purposes, okay, untuk makan, okay, we use the young one. Okay, the same goes to the nasi ulam. We use the young ulam for our food because I think the taste is better. Okay, lebih enak. Kalau lebih enak if you use the young inflorescence. Okay, and scientifically, okay, the young inflorescence has active tissue compared to the old one, compared to the blooming stage tissue. So for sure, if you want to study the biochemicals, we use the young inflorescence because they contains a lot of active biochemical compounds during the early stage of the inflorescence. Okay, I think I have answered two questions, and if I'm not mistaken, I got another question from Siti Fatima and Mr.
Sorry, Dr. Yunus. Maybe we can give the chance first to Miss Vina or Miss Faya. Maybe they have feedback from the question. Yeah.
Okay.
Miss Vina, do you have any feedback, or is it enough? I think is it enough? Miss Maya Melati. Do you have any feedback? Okay, enough. Okay, let's continue to the. But before continue to question for Dr. Yunus, we will give the chance to question for Professor Sinsi first.
Okay, yeah. Thank you.
Yeah. Thank you. Pak Musafan, please, the question for Prof. C from Mr. Gilranger. I just addressed my question about sorghum. It seems that hybrid sorghum more vulnerable to disease compared with the inbred variety. It is just one question. Yeah, please, Professor Sid, you can directly answer this question from Mr. Chiu. Oh, there are two. There are two questions. The next question is from Miss or Mr. Dwi. Have your team had the transgenic or the candidate sorghum plant based on CRISPR? Please, Professor Sinsi. Yeah.
Okay, thank you. That's good questions. For the first question, the question is why does hybrid of sorghum still met a lot of problem or conquered by the disease? I can answer because as we know, the pathogen can mute very frequently than the plant. If we made a plant, a hybrid plant over 5 years or 10 years, but during the large scale growth, we grew in the field with a large scale, and a bunch of pathogen will conquer the maybe in the first stage or first several years it's very resistant, but during the later period, because just like our COVID-19, and this virus is continue mutant, and the mutant because the targets are lost. So I can say it's like a wall. Can know the wall between the plant and the pathogen is continue forever, and no which one is the winner. And I think the first question is I answered. And for the second question, so about the transgenic line of the sorghum, I am trying to do the transfers candidate genes in the sorghum plant, but still now unsuccessful because it's hard to do the transgenic plant in sorghum. And we tried to transfer some genes, candidate genes in the rice or in the tobacco to verify the functional. And in the last, what we want to know the function in the sorghum. And I also know some labs are successful. Can they successfully transfer genes in the sorghum? So and that's all. It's my answer.
Okay, thank you. Pak Jill, do you have feedback for the question answer for
for Dui? Yes, I. Okay. Also, okay. As I will also text in the window.
Okay. Thanks, Professor. I have been visited China and I read some place that cultivate a lot of sorghum. That's why I asked, because I am from Timor-Leste. We have been introduced also some inbreed lines that's coming from India. That's why we distribute, we release to the farmers. I think that's we have facing some problems in there.
Yeah.
That's why I asking. Thank you for your explanation.
Okay, thank you.
Okay, thank you, Pak Cil.
Yeah.
And then from Miss or Mr. Dwi, do you have a feedback?
Yes,
No? Okay. Yes.
Yes, I finished this question. I answered this
Yeah,
question. Yeah. So
okay, I think it is.
if I have more questions, yeah, we can interact in the test.
Okay. Thanks, Prof, for the answer and for the informations.
Okay, thank you.
I think it's enough.
Okay,
thank you.
Yeah. Okay, we'll continue to the next question to Dr. Yunus from Siti Fatima, Miss Siti Fatima. Is the chemical compound of torch ginger influenced with anti-abiotic factor such as soil type, elevation, or etc.? And then the next question is from Mr. Hakim Kurniawan. As torch ginger is planted by farmers as small farm in Malaysia, what would be the main challenges in propagating and cultivating it at large scale? Thank you. Please, Dr. Fahmi Yunus, you can give the response for these questions.
Okay, Assalamualaikum. Thank you very much to Siti Fatima for the questions. Okay, your question is about the chemical compounds of the ginger influenced with abiotic factors such as soil type, elevation. Okay, actually, to be honest, it is a tough question for me. Okay, at the moment, to the best of my knowledge, there is no report about the study of abiotic factor, soil type, on elevation on the chemical compounds of the torch ginger. So it is to be safe, I do not have any answer for your questions, but actually it is a good question, and maybe other researchers can do this type of study about the relationship between the abiotic factor and also the soil type, elevation on the biochemical compounds. Previously, researchers in Malaysia, they do not study the abiotic factor, soil type, or elevation. They just study different states and they relate it with the chemical compounds. And if I'm not mistaken, it same goes to the Indonesian researchers. They have published a lot of publication in 2020 and 2021 about the chemical compounds. And same as like Malaysia, they do not link the chemical compounds with the abiotic factor. Okay, maybe Siti Fatima, you can have a study about it. Okay, this is like a good question and it opened a new path for the research. Okay, and the second question is for Mr. Hakim. Okay, Mr. Hakim, thank you for your questions. Okay, what is challenges in propagating and cultivating it at the large scale? Okay, I'd just like to remind everyone that the torch ginger is an underutilized crop. So you know it is underutilized crops mainly propagated in kampung, in village in Malaysia, but still have like a small scale farms of the ginger in Malaysia. And the most challenging part about the large scale cultivation is about the land availability, the agricultural land in Malaysia. I think you know. Okay, and the first thing about the land availability or the land for agriculture is competition with the well-known plants such as, I think you know, the oil palm, rice, paddy, and for sure about the other vegetables that can be grown in a glass house. So you know that's some of the ornamental flowers like rose, carnation, orchids. So it is easy to export them. So the most of the farmers prefer this kind of plants. Okay, the oil palm, rice, and different type of vegetables and ornamental flowers. And the second one is about the challenging is about the market. Okay, most of the young inflorescence that we harvested in Malaysia, they use it for the culinary purposes. I think for food, yes, for culinary, for food. So for sure, if you want use it as an ingredient in your food, you need a very fresh inflorescence. So, but the problem at the moment, like the young inflorescence, they have a short vast life, and maybe it will be rotten. in just 6 days. So, you know, it's from the village that you need to bring it to the town. So it takes time and you need to sell it as soon as possible before it turns into brown or black because of the rotten processes. So that's the second challenges in Malaysia. Okay, but so that's why I think researchers in Southeast Asia, in Thailand, Malaysia, and Indonesia, I think they are trying their best to move these plants to more high-end products such as the cosmetic products. And also in Malaysia, they use it as a part of the reducing agents in nanotechnology. So we can use it more frequently because this plant is totally available throughout the year and it is easy to propagate it actually. For the money or the seed money for the business for Malaysia, for the agriculture, for the young agriculture farmers, they do not have any problem with loan from the government, and actually it is a good opportunity for them to take the government loan and to use it for the large scale of propagation in Malaysia. Okay, is it okay, Siti Fatimah and Mr. Hakim?
Okay, is there any feedback from Bu Siti Fatimah or Mas Hakim?
Thank you, Dr. Yunus, for the answer. I think it's clear enough. Thank you.
Okay, thank you to both of you.
Thank you. Okay, and then we will move to the next question, but we don't see in the chat room their question. So I will give a chance to all the participants to give the question, and especially maybe for the Professor Wang Yang. Maybe any question for Professor Wang Yang? Any question? You can directly give the question in this Zoom room. Any question?
Question.
Not yet. Okay. Maybe more question for Dr. Fahmi Yunus or Professor Sinsie.
Yeah.
Oh, okay.
Yeah.
I think this there is a question here from Ms. Tita Wijayanti to Dr. Yang Wang.
Yeah.
Thank you for your nice presentation. I want to ask what the most common disease with the highest severity in Guizhou, China. Please.
And in Guizhou province, we have the many cancers. I just say mainly the leaf disease is and for the cereals, and the leaf disease is most serious. It of course it's caused by different kind of the pathogens.
Okay.
Yes.
Ms. Tita, is it clear enough for you?
Yeah.
Okay. Yes.
Yeah.
Thank you very much. I will give one more chance.
Okay. Yeah.
I will give one more chance to participant. I will give a question for our three speakers. No?
Yeah.
Okay, as there is no question, we will finish this session. Once again, we thank to three speakers from the first session today. But before we finish this session, there is a gift from us to the speakers. We have certificate for you for our appreciation for all of you, the speakers. Mas Adit, please share the certificate. This is the certificate for Dr. Muhammad Fahmi Yusuf. Thank you for your presentation today. And then the second one is for Professor Sinshi. This is certificate for you. And the third one is for Dr. Yongwang. We will send this certificate through your email address. Yeah. Once again, thank you very much for your time and for your all of information about the underutilized crop. And we hope that we can meet again in other occasion. And let us give to all of the speakers big applause. Once again, thank you very much. And I will give back the question to the Master of Ceremony. To Mbak Dian, please.
Thank you, Mr. Darmawan. Thank you for all the invited speakers and also our moderator today, Dr. Darmawan, for an amazing session 1 presentations. And then we still have another sessions, the second sessions. And let's to take the advantage of time, let's start the second sessions with the invited speakers presentations. And for the sessions, it will be led by our moderator today, Dr. Afifuddin Latif Adirejo, SPM SC. So for Mr. Afif, time is yours.
Okay, thank you, the Master of Ceremony. Just check first. Is it clear? My voice from here?
Yeah, is it clear enough, Afif?
Okay, thanks.
Mr. Afif, but excuse me, we still have a video to introduce you to the audience. So please kindly wait. Okay, Dr. Afif, time M4 is yours.
Okay, thank you for the Master of Ceremony. Dear ladies and gentlemen, I'm pleased here to be a moderator from this last plenary session, I think. Well, we have again 2 experts to present in these sessions. Before we start, I just want to make sure if our honorable presenter have already here. Hello, Dr. Prakit Somta. Are you here in this Zoom meeting? Just want to check first. Or maybe I will call first Dr. Noarwe Christians. Hello. Maybe I need some help from. Hello.
Noarwe Christian is ready.
Okay. And then how about Dr. Prakit Somta? Maybe I need some help from the official committee. Please check for the availability of Dr. Prakit Somta. Okay, I think there is some problem connection maybe from Prakit Somta. For all participants, normally the first presentations will come from Dr. Prakit Somta, but like you have already here, Dr. Christians, who has already joined in this forum, I will let you present first. And before we start, please, official committee, announce the curriculum vitae of Dr. Christians. I will read some of biography of Dr. Christians. Please, the official committee, for the curriculum vitae of Dr. Noarwe Christian from Nigerian Institute for Oil Palm Research. Hello, check. Okay, thank you. So normally Dr. Noe Christians will present for the second presenter, but I will call first you for the first presenter. Dr. Noe Christian from Nigerian Institute for Oil Palm Research. Hello, sir. Good morning from here. The complete name of Dr. Noe Christian Ngozi from Nigerian Institute for Oil Palm Research. He will present the interesting topic in this Zoom meeting, harnessing the potential of sea butter plant in the Nigerian economy. I will remind you, Dr. Christians, you will have 40 minutes to present in this Zoom meeting, and then we will have discussions after the presentation of Dr. Prakit Somta, normally like that. I hope Dr. Prakit Somta will join after your presentations. Okay, time is yours, starting from now. Please, Dr. Christian. Is it okay, sir?
I'm finding it difficult to upload my document.
Okay, thanks.
I'll get back to you immediately. I'm able to upload my document, sir.
Yeah. So
I'm trying to upload my document. Please give me some time to attach my document for presentation.
Yeah. So before
you can go on with
your... before hearing and what I will say, participating in your presentations, I will inform the audience here that you are researchers in Nigerian Institute for Oil Palm Research, and here in the biography you say that your experience in research and development span over 2 decades and 2 years in economic of tree and food crop, especially oil palm, coconut palm, date palm, raffia palm, seed tree, and farming system research. And you are Director of Research Agricultural Economists in Nigerian Institute for Oil Palm Research, or NIFOR, and your activities include development and execution of multidisciplinary research project proposal, resource person in consultancy service to different stakeholders. Okay, it was good experience, I think, and then we will curious to hear what your experience, especially concerning the topic that you will present today. Just want to say hello, if maybe Dr. Prakit Somta join to this Zoom meeting. Hello, Dr. Prakit.
Yes, I'm in the meeting now.
Okay, sorry, because when I call you, when I open the sessions, I didn't hear anything, so I gave the first sessions or the first presentation to Dr. Christian. Is it okay for you? Or how do you think? For me, wait. Yes,
actually,
please.
I was engaged in another meeting, so if possible, I want to present now. It's okay?
Okay, no problems. So is it okay, Dr. Christian? Because Dr.
It's okay.
Okay,
It's,
very good.
It's
yeah,
okay.
Yeah.
It can present while I sort out myself.
Yeah.
So,
Okay. Thanks.
okay,
So,
thanks.
but first of all, Dr. Prakit, I will explain first the brief biography of yours. Please, the official committee, let participants know concerning Dr. Prakit Somta. He comes from Kasetsat University. In his biography, he wrote that the interests of research include resistance to disease, especially leaf blight, and days to maturity. Currently, disease-resistant crops are available, approximately 450 seed per cross, and also crops for days to maturity. So I need some help from official committee. Please let participants know the biography of Dr. Prakit Somta, because Dr. Prakit Somta will present for the first presentations.
So I can present now?
Wait, sir. Just. Or it's okay, yeah, because you have a short time. It's okay. I think I have already read your CV or your biography.
So
and
I can start now, right?
Okay. Please, starting from now, you have 40 minutes. Okay, I will remind you in 35 after 35 minutes, more or less, right?
Okay.
Okay. Please, time is yours.
Okay, so I cannot share the screen now because
it's not possible.
No. So
please, the official committee, the host, I don't know who handled this, Mas Adit or Pak Fuad, please help Dr. Prakit Somta, because he cannot open the screen due to the possibility of co-host, I think. You can try again, Dr. Prakit, if you have some difficulties.
Dr. Prakit has already become the co-host, so
okay.
You can see my screen, right?
Yes.
Yeah.
Okay, thank you.
Okay.
So please wait.
Yes.
Some problem.
Everything is okay, I think. It works now.
Okay, so. Okay, you can see the screen now?
Not yet.
Not yet.
Unfortunately, not yet.
Okay. Okay, this one. Okay, I think you can see this.
Yeah, good. So I'll give back to you.
Okay, thank you.
Time is yours now. Yeah.
Okay, all right.
Yeah, please.
Okay, thank you, the moderator, and thank you the organizing committee of this conference for inviting me to join this workshop and give me the opportunity to present my work. Anyway, I like to change my topic a little bit because I inform the committee that my topic is about wing bean. But today I like to present my work on lab. But the topic is really similar. That is the genetic diversity of lab gene pool in Thailand. So why lab? Lab is an ancient legume crops of Asia and Africa. It has been domesticated about 3,000 to 4,000 years ago in Africa, and then it was introduced to Asia, and is now widely grown in Asia, especially in Southeast Asia. I think in Indonesia you also have these crops. In Thailand, it's widely grown in the rural area. It has a high potential for commercial production. The young seeds and young pod consume, and currently, as it is underutilized crops, no breeding program in Thailand. So most of the all not most all of the cultivar grown in Thailand. I think in other countries too, they are landless mostly. But in China, maybe they have breeding program, and also in India, but it's a small breeding programs. And the origin and domestication place of this this crop, I mean the lab, are still unknown. Still debating about the origin and domestication of these crops. So I am interested in these crops, especially because in Thailand, no study about the genetic diversity of the lab in Thailand. So you can see this. This is the lab. You can see that it has a high yield potential. So in terms of utilized crop, it can improve into the commercial production. You can see the wide variation in the pot characteristics of the lab. And this is the lab pot. So in the local market in Thailand, you can see wide variation of the pots. Mostly in Thailand, we consume the young pot and the young seed, but also in some area, people also consume the mature seed. This is what I mentioned, the seed, the young seed also consumed, and also the sprout in this is in Myanmar. This sprout from lab seed. It's really interesting. We in Thailand we don't consume this. I mean the sprout, the young sprout, very young sprout of the lab. The dry seed. This is in Myanmar on the left hand side, and it's we also consume the dry seed as a snack. This is in Thailand, and some local producer try to develop some products from the dry seed. This is from India. You can see really wide variation to of the pot too. Some oblong, green, some purple, some flat and wide pot. This is you can see the variation in the pot. According to the pot characteristics, lab slab can be classified into 3 subspecies. One is Ancinatus. This subspecies has a short pod with 2 seeds per pod. The other subspecies is Purpureus. This species have a longer pod than Ancinatus, flat and wide pod with 3 to 4 seeds per pod. And Bio-Industry, this subspecies has a linear or oblong pod with up to 6 seeds per pod. So this is a proposed genetic relationship between among the 3 subspecies Ancinatus, Purpureus, and Bio-Industry of the lab proposed by Dr. Masses. The 2 seed pod, I mean the subspecies Ancinatus, this subspecies are present in Africa. In Asia, this subspecies does not exist. And the other subspecies that had 4 seed pod is the subspecies Purpureus and Bio-Industry. These 2 subspecies widely grown in Asia and Africa, and some in India also. The objective of my study are to explore and collect the lab germplasm in Thailand, and then assess genetic diversity of the collection, the lab collection. Mostly the lab is grown in the north, the central region, and the northeast and the east of Thailand. So we have a survey trip, the exploration trip, in the north, in the west, in the central, and in the northeast of Thailand. About 25 or 30 province in Thailand. And this is our exploration trip and correction trip. Mostly the lab in Thailand is grown by farmers as garden crops in the backyards or in front of in front of their home. This is my student. They were collecting the seed or the leaves. When we collect the sample, in case we cannot collect the seed, we collect the leaves, and we use the leaf for DNA extraction and genetic diversity. You can see in Thailand, most of not most, or actually all of the lab cultivar are really rigorously, and it's really high. Is a climbing. You can see it's grow up to the tree. This is variation of the pores of the lab in Thailand. This is a subspecies Papilius, and this is a subspecies bengalensis. In Thailand, we have only two subspecies. We don't have Ancinatus. Some farmer they grow more than one type. I mean the subspecies and subspecies Papilius and subspecies bengalensis together in the garden. From the correction, we collect leaves from 229 individual plants, 145 from the north, 113 from the northeast, 11 from the west, and 30 are from central. And we extract the DNA from this individual, and then we use SSR marker to determine the genetic diversity of the lab. At the beginning, we screen 145 SSR marker from lab, from cowpea, aspy bean, mung bean, and common bean in 6 lab individuals. You can see this. Most of the markers can define the DNA of the lab, especially the marker from different species. More than more than 70% of the marker from other species can amplify the DNA from the lab. This indicate that the genome conservation between this Vigna species and Fasciola species and the lab are highly conserved. However, the polymorphism of the SSR is not high. Allowed in average is about 12%. And then we determine the genetic diversity of the 299 individual. And the number of alleles vary from 2 to 4, with the average of about 3. And you can see the observed heterozygosity is really low. Actually, we didn't expected this because the flower size of the lab is big, so we expect more. We expect more outcrossing, but according to the DNA marker analysis, the heterozygosity is really low. It's about 0.03. And gene diversity or expected heterozygosity is about 0.36. This is relatively low, and of course the polymorphism information content of the marker is low too. And then we determine and compare the diversity of the lab that come from different regions, but we found that in most cases the number of the diversity among the region are comparable. If we look at the allelic richness and the gene diversity or the expected heterozygosity, the lab from different regions have comparable variable of the allelic richness and the gene diversity, and the fixation index is really high, nearly to 1. This is agree with the observed the observed heterozygosity that is really low, and we estimate the outcrossing rate. The outcrossing rate, the average is about 3.3%. The maximum is about 5.4% in the west of Thailand. And then we compare the diversity between the subspecies Bengalesis and the Piphilis. You can see that the diversity between these two subspecies is really similar, except the outcrossing rate in the Bengalesis that is about twofold higher than the subspecies Piphilis. So this means the diversity between these two species are almost the same. Then we determine the genetic structure of the 299 individual. The population are separate into sub populations, and then we do the phylogenetic tree. The neighbor joining tree to determine the genetic relationship among the individual. You can see that individuals from different regions are clustered together. You can see this is from yellow from the north, green from the northeast, red from the west, and blue from the central region. You can see that we cannot group them based on the origins or phylogeny or geography. And then again, this is a neighbor joining, but this time we look at the relationship between the different subspecies, the Piphilis and Bengalesis. Again, you can see that these two subspecies are mingled together. We cannot separate them based on the pod characteristics or the subspecies. This is PCA, the same result with the neighbor joining tree. The individual or sample from different regions cannot be separate, or no pattern or no clear grouping among the among different regions. And also, the PCA show that the germplasm could not be grouped based on the pod characteristics or the subspecies. You see that they are mixed together. So in conclusion, I conclude that the genetic diversity of lablab in Thailand is relatively low, and the population of Thai lablab is unrelated to geography, nor related to pod characteristics or the subspecies. So I would like to thank Kasetsart University in Research and Development Institute for funding this research. And we have published this work in the breeding science. If you're interested in this work, you can follow, or you can read this paper for more information. Okay, thank you for your attention.
Okay, thank you, Dr. Prakit. It was very good presentations. I think you have explained some substantial things, but you spent only about 17 minutes, so you have many times. But before going to the second presentations, maybe you will have some additional informations along and besides of your slides. Please, if you have.
Pardon?
Maybe if you have some additional information that what you like to explain more. No?
No, thank you.
For the moment, it is all, and we will have the second presenter.
Okay.
Like okay. So thank you, and then we will discuss after the second presentation from Dr. Christians.
Okay.
Dr. Christian, are you ready for the second presenter? Hello, Dr. Noit Christians. I will invite you to present after Dr. Prakit Somta. If maybe one of official committee can help, please give some assistance to Dr. Noit Christians. I think he got the problems due to the connection, or maybe some difficulties of joining the Zoom. I don't know what kind of problems. Hello, Dr. Noit Christians, can you hear me?
Mr. Afif, I'm sorry. I think that there is a problem connections with Dr. Nuawe. So we're waiting for him to rejoin.
Is he still here, Budian? No?
No,
I will search in the list of participants.
No, he is not here yet. So I think that we can start to do the discussion first while waiting for him to come to rejoin the Zoom. Meeting.
Yeah, maybe if there is some communications outside of the Zoom, please contact Mr. Nuawe Christian if possible for the official committee. Thank you in advance. I will invite again, sorry, Dr. Prakit Somta. Are you still here? Dr. Prakit? We are sorry for this inconvenience, especially for the audience and all participants here due to the problem of the connections. This is one of disadvantage of the Zoom meeting, especially also for Dr. Prakit Somta. If you have time directly to answer the questions, hello, I will make you sure first if you are available to answer direct discussions. Hello, Dr. Prakit Somta. Can you hear me? Or maybe I need some help to the official committee. Okay, I got some information from the committee. Dr. Prakit, just take break first. Okay, now I'm going to invite all of the audience, all of the participants to ask some information, maybe to ask some things that have just presented by Dr. Prakit Somta. If you have some questions, please raise hand, or maybe you can write in the Zoom chat, please. Okay, this is, I don't know if the official committee, would you like to help me to present the questions, or I have to directly read here? Okay, might drop your questions. Okay, nice presentations. Okay, this is the first question, I think, from Siti Fatima Hanum. Thank you, nice presentations. I'm wondering how Thailand government conserve underutilized crop. Okay, we will wait for Dr. Prakit Somta. I will invite again, please the audience, please participants, if you have some questions, don't be hesitant to ask in the Zoom meeting. You can directly speak orally if you want, but if you don't, if you choose to write, it is okay. Yeah, please write in the chat room. The official committee will rewrite in the slide, I think, after that. Pak Musafan, if possible, please the slide of the questions.
Mr. Afif, I think that Dr. Nawawi have already rejoined the
Okay, very
meeting.
good. Yes, thanks for your information. Dr. Nawawi, are you here now?
Yes.
Okay, very
now.
good.
Let me try to share the screen.
Yeah. It was problems before?
Yes.
Okay, it's okay. Yeah. So before starting, I will remind you, you have 40 minutes, if possible. You can use all 40 minutes. You can start from now. But I didn't see anything from, okay. So starting from now, time is yours. Please, Dr. Nuawe Christian.
Thank you for giving me the opportunity to be in this this conference. I will quickly go through my presentation on the underutilized crop with respect to share. In 2020, Nigeria's GDP amounted to 152.3 trillion naira, which is equivalent to over 40 billion US dollars. Out of this, Nigerian agricultural sector, that's about 24% of the GDP, was put into crop production. This accounted for 21% in 2020. The share subset accelerates the pace of economic development of the Nigerian economy, if adequately harnessed. Therefore, it is imperative to constantly maintain the flow of information, production, export, and import area under cultivation of the industry. The challenge with this share tree is that there's paucity of information across the whole country. It is in the recognition of the aforementioned that the federal government of Nigeria mandated the Nigerian Institute of Oil Palm Research, NIFO, to carry out research on the share tree. Agricultural production must be seen as a critical component program that Saharan African countries in Nigeria, Mali, Burkina Faso, Ghana, Benin, Cote d'Ivoire. The major producers of shea nuts in the world the world's average yield of shea nuts from 2000 to 2019 was put at 639,640.26 metric ton. Out of this, Nigeria accounted for more than half of the world's shea nuts production. For over 10 years period with an average of 358,003.1 metric tons. Nigeria is the largest producer and has the shea nut producer. Nigeria is the largest shea nut producer in the world since 1990 to 2019. Now, if you look at what I'm showing you here, this figure shows the distribution of the shea tree in Africa. The shea nut production in Nigeria showed a progressive increase from 88,000 metric tons in 1975 to more than 490,000 metric tons of the entire shea producing belts in Africa. This is the production situation across the African countries. And you see what Nigeria commands with what I show you here, production situation. This is also a figure showing the distribution when you compare Nigeria to other African countries and see where Nigeria stands. Now, shea tree yield. The shea tree can attain optimum yield of up to 45 kilograms per annum. Some can produce between 50 and 50 kilograms per annum. The kernels of the shea fruits are made up of 40 to 50%. The dominant species of shea tree in Nigeria is the Paradoxia, West African kind. According to Dandy Forest Seed Center, the species produces more solid shea, but contains more stearic acid than the Viteria nilotica species. Nigeria has the largest shea tree density, the largest producer of shea butter in the world. The production data continues. In spite of what I am presenting here, and the massive potential that Nigeria has, this commodity is still highly underutilized in Nigeria. This is export and import situation. Shea products have lately assumed an unprecedented height in the economic growth. Shea nut and shea nut butter are value-added products with outstanding export growth potentials for Nigeria and other African countries. Nigeria earned $54,000 from shea nut export in 2016 and exported over 50,000 tons of shea butter annually in 2018 to 2019. It is believed to account for almost 60% of the world's supply of shea butter and allied derivatives, valued at about 3.8 billion every year. The shea butter industry in Nigeria is dominated by small-scale production plants with production capacity of less than 0.1 tons per day. The leading producer of shea butter in the world, with a production capacity of about 500, with a production capacity of about 500 metric tons, produced mainly from the use of crude implements, is yet to fully realize its potentials in the processing and export of shea butter. Shea kernels have been substantial economic importance to Nigeria, as they consist of over 55% oil. Establishment of shea plantation has the potential of boosting the farmers' productivities, which will as well contribute to the nation's foreign exchange earnings. Shea kernels have substantial economic importance to Nigeria, as they consist of 55% of oil. Establishment of shea plantation has the potentials of boosting the farmers' productivities, which will as well contribute to the nation's foreign exchange earnings. The establishment of shea plantations and promotion of export will not only add to the total economic GDP of Nigeria, but will invariably make positive impact on the income of the citizens, as well as the diet of the people, thereby contributing to the standard of living of the entire population. Uses. Shea butter is used in the cosmetic industry for its high power, and is often needed as a substitute for cocoa butter in the chocolate and confectionery industry. Almost every part of the tree has economic importance. The fruit could be eaten and the leaves are used as fodder and serve as an ingredient for making alkaline and paint. Shea nuts and the activities generated as a result of the production of shea butter act as a source of livelihood among the rural, where it is predominantly available in the wild. The value chain. Shea value chain represents a major source of income. It is a weapon against poverty for both rural women and youth, through employment generation and wealth creation from their diverse practices along the value chain. The chain includes collection of shea fruits, processing of shea nuts and butter, firewood, gathering, charcoal production, as well as mortar and pestle making. These are economic activities, these are economic ventures in the shea value chain. The shea nuts collectors and shea butter producers are the main chain actors. Others are just actors along the value chain that contribute negatively to the sustainability of the shea tree. Shea fruit collectors, gatherers are the first group along the value chain, and they are sometimes referred to as nut traders. They are majorly involved in shea fruit collection and processing, and they occasionally process the nuts into butter for domestic uses and sell in the local market. The collection and subsequent processing as butter or sale for local women. This is part of the shea kernel, a robust shea nut. For those who don't know what this commodity is all about. Now, the shea butter producers are the pivot of the industry. They add value to the kernels to produce shea butter, which is an important component gotten from shea trees. This is a typical example of shea butter. Now, the shea tree is increasingly being used for its wood Buyers mostly demand the wood. They believe it is robust, lasts longer, burn with great heat, and is cheaper than other energy sources. This has a devastating effect on the shade tree density, as trees are illegally and persistently being sliced into wood logs or tied in bundles of sale. In Nigeria, the demand for fuel wood is very high because making it into the in Nigeria, the demand for fuel wood is very high because more than 80% of the households use firewood for their cooking, making it the most used form of cooking energy. Though people earn income by gathering wood from share, it however destroys the resources that provide the rural populace with substantial income. This is how this share tree is being logged in Nigeria for firewood. Share tree is believed to produce bigger and stronger charcoal. For business of trading in charcoal has become a very lucrative in Nigeria. The production, transport, and consumption and combustion of charcoal constitutes critical energy and economic cycle in the economy. About $170 to $300 per metric ton is gotten from share tree from those who trade in this. The charcoal trading business is a lucrative one for anyone involved, whether as a retailer, wholesaler, or supplier. In most parts of the country, charcoal is the primary energy source for cooking and heating, as well as major source of income generation and environmental degradation. This is charcoal made from share tree. Ogunsanya observed that tree species such as Malaysia, Etlingera, and Commodity are used for making mortar and pestle in Nigeria. Mortar and pestle making, sometimes classified as part of wood carving, is an important economic activity. The logs used for mortar and pestle making are harvested indiscriminately from the wild, from both mature and young trees. The challenges. In Nigeria, we have declining tree population. Share tree populations are under threat for a variety of reasons, including lack of new planting, reduced fallow periods, poor parkland management practices, and tree removals for charcoal production or due to mechanized agriculture. Lack of new plantings. Share trees are rarely planted due to long gestation periods, cultural issues, and lack of field experience. Long-term availability of supply. The demand for share tree has grown to over 600% in the last 20 years, and the industry forecasts a future 50% increase in the next 5 years. While tree populations are declining at a population density of 0.5% per hectare annually, if current trends continue by 2034, there will be a supply shortage in West Africa. Climate change. Yields are negatively impacted by climate change. Decrease in the population also reduced farmers' resilience to floods and droughts. Improving tree population is a key for the supply chain to act as catalyst for growth in Nigeria. In conclusion, the federal government of Nigeria is making desperate effort towards developing the Nigerian share industry. Recently, a national initiative known as Spare Nigeria Share Parkland Afforestation and Reforestation Nigeria was introduced with the following interventions. Establish scalable model and create transformational change by changing the incentive systems. Conclusion continued. Yes?
Please, official committee. Okay, no, please continue. No, it's okay. I'm sorry. We are sorry, please.
Okay.
Yeah.
Now, conclusion. The federal government of Nigeria is making desperate effort towards developing the share industry. Recently, a national incentive known as Spare Nigeria was introduced with the following interventions. establish a scalable model and create transformational change by changing these incentive systems around protection in share producing communities. The project will regenerate parklands while ensuring that communities have short-term economic benefits from their involvement in tree crop value chains. After 5 years, the project will achieve the following objectives: rejuvenate 200,000 hectares of share parklands, facilitate 600,000 metric tons of share sales, improve climate resilience of 100,000 women share collectors and their youths. Specifically, the project will deliver the following outcomes: 10 million trees planted, 100,000 women and youth trained in parkland management, and set up to engage in commercial seedling farming and parkland management. State legislation against tree cutting in the project areas. Better community awareness of economic value share trees and need to protect them by in and ownership of the preservation of growth of the parklands initiative, fueling active and sustained participation by communities. Conclusion continued. Other interventions include assessment of current tree population and spread in Nigeria, large-scale bursaries, private sector participation, parkland management techniques to protect existing stock of trees, state level legislation against tree cutting, awareness creation, sustained communication, and community engagement. To this end, these initiatives are expected to serve as key to unlocking the potentials of the share industry, and also as an impetus for its growth and development. Thanks for listening.
Okay, thank you very much, Dr. Noe Christian. It was very valuable informations. Now I'm inviting the audience or the participant here to ask something for our honorable and our expert, Dr. Prakit and Dr. Noe Christians. Just want to check again, Dr. Prakit, have you already joined the Zoom? And please also take a pins in the Zoom for the committee to Dr. Christians, if possible, 3 persons in the Zoom. We have 20 minutes, more or less, for the discussions. I read some questions before, so now we will have the second sessions of these plenary sessions. In waiting another questions, I will directly read here the first question from Siti Fatima. I think Siti Fatima from Madura Islands. Yeah. So the questions for Dr. Prakit. I'm wondering how Thailand government conserve underutilized crops. Yeah, time is yours. Directly, please, Dr. Prakit.
Actually, this question is really difficult to answer. Actually, the government has no clear policies regarding the conservation of underutilized crops. For example, the lab. I think the Department of Agriculture, I mean the Ministry of Agriculture, they don't do the correction or any conservation of lab. So this is a big problem in Thailand too. You know, in Thailand, we the government only focus on the major economic crops they don't care about the underutilized crops.
Okay. Before asking again to Siti Fatima, so just need more confirmations. Is there any gap between the government and the university's policies, like you, for example? How can you and your college as a researcher or lecturer can match this kind of gap? Can you understand what I mean?
Yeah, I understand. It's really difficult
Okay.
they don't, I mean, the government, they don't realize or knows the potential of the underutilized crops.
Okay, I see. Yeah. So I give back again to Siti Fatima. I don't know how can you give me feedback, because you wrote in the chat, but if there is no feedback in the chat room, I think the answer is enough. Yeah. So the second question is also from Siti Fatima, I think. How long the longest life of lab plants in Thailand, and how long is the productive period? So please, Prakit.
Usually, since most of the labs cultivar in Thailand are landless, they are sensitive to their length. Usually, they will produce a flower and pot during the short day condition in Thailand, starting from October until December. So the farmer goes the lab starting from they grow in the rainy season starting from August, and then the lab will flower or set pot in October or something. The lifespan is about 4 or 5 months, depend on the cultivar, and the reproductive period is about 1 to 2 months.
Okay. Sorry, for Siti Fatima, will you give some feedbacks if you want? You can write directly in the chat room. Okay. I'll again invite all of participants. Yeah. Now please ask if you want to ask to Dr. Prakit or Dr. Christians. You can directly. Okay. I read here some questions from Maya Melati, yeah, to Dr. Christian. I'm Maya from IPB University. So Dr. Christian, IPB University is one of the biggest university in Indonesia in the domain of agriculture, yeah, just like University of Brawijaya. So CNAT, yeah, I think this is CNAT. You miss A, yeah, okay. CNAT has many valuable use, but unfortunately, it is becoming rare and extinct. Yeah, it will be left extinct or any effort to preserve. So but I will try to reformulate the questions from Dr. Maya Melati. So how do you think, Dr. Christians, concerning the diversity and the cultivations that become more and more rarely, yeah, especially in your country? Please give some answer. Time is yours, Dr. Christians.
This, I didn't get the question. I didn't get the question correctly.
Yeah, I see. So I think so that's why I help and I assist the questions. It means what is your solutions or the solution of your government, for example, if you have this kind of challenge, for example, the extinctions, or that you call maybe the destroy, right, in your slides, the destructions of this kind of plants. So what what will you do in the future, starting from now, for example?
Okay. Thank you for this beautiful question. In Nigeria now, there is a lot of awareness going on. Right from the Nigerian Oil Palm Research that has mandate to improve the growth and productivity of this shea tree. Now, they are collaborating with the state governments where you have these trees dominance, creating the awareness why the destruction of these trees has to stop. Because apart from the environmental impact that it causes, if there are a lot of advocacy trying to get people on board on how they can really go on improving on the processing and marketing of what is available, that more income can be gotten from engaging in better activities than cutting down those trees. The parklands will be protected. And like I mentioned in my presentation, I said the federal government recently has a national initiative known as the Share Parkland Afforestation and Reforestation that is embarking on, which was introduced with the hope that there will be a lot of education going around most of these communities and educating them. And this is already going around the whole country where these trees are supposed to be cultivated. I hope I've answered that question correctly. Hello?
Yeah. Okay. Especially for Maya Melati, if you want to say something directly in the Zoom, please, I will give you time. Maybe you have some feedback or want to ask some information against concerning your questions.
Yes, I understand it's not easy to conserve the plant that, with different uses. Yeah. For example, as I heard that there are also used for from the wood, so they harvest the stem or something like that. So I understand that's a challenge. I have no suggestion, but I'm sorry that I have no suggestion how to conserve the plants, but it's a big, it must be from the government and also the community to understand how important this.
Sorry, I interrupt you. Please, please committee for the picture of Ibu Maya Melati here. So I think for person who speak directly, please give pins for him or her. Yes. Okay. Directly, Ibu, please.
Thank you. Thank you. Yes. I just we wish you that you will be allowed to conserve the CNAT, because I know that this is very important plant, but I know that there is a problem in the community, and also I understand that this is the problem in the community that harvest because the plants, I know that the tree have many uses, so I think that's a competition between the CNAT, the fruit and also the stem, I think. So I hope you, the government will be allowed to conserve these plants. Thank you, Pak Asif.
Thanks, Miss Maya Melati. So before going to Dr. Christian, so Miss Maya Melati, are you concerned in this kind of plan, or maybe just want to ask deeply, or how about your activity now concerning this kind of species?
No, we don't have it in Indonesia, maybe.
Yeah. No, I understood, but I think it will. I mean, how did you do before, maybe in your experience, for example, because I didn't knew personally, we don't know before, but I mean, if you have concern of this kind of species, maybe abroad or in another countries, yeah, you know, because this kind of species growth in several countries, especially from Africa, the countries Mediterranean, even in Europe, for example. So that's why just a simple question for you. No, just in the context of knowledge, you ask this species.
Yeah, just
okay.
the concept of knowledge, because I just heard it from this seminar, so I have a lot of information. It's good for me. It's enrich my knowledge about underutilized crops.
Yeah. The problem is similar like our oil palm, I think. Yeah, you know, this is exists the competitions between the need for the consumption and the need for the fuel, for example. But this kind of plant fuel directly fuel woods. Yeah, but the oil palm maybe we call fuel is like biogas or bioethanol, etc. So the similar problems exist like oil palm and this kind of species. Okay, it was very beautiful, like the comment of Dr. Christians, beautiful Christians. Okay, thanks. So I think it was okay your answer, Dr. Christians, concerning the question of Ms. Maya Melati from IPB University. I come back again to chat room here. I don't see now the questions. Okay, this is a commenter, I think, just opinion. Okay, from Muhammad Madni. I'm Muhammad Madni, Ph.D. Forestry Research Scholar at Department of Forestry Range and Wildlife Management, Pakistan. Okay, I think this is just opinion. So I'm inviting again, please, all participants who would like to ask some questions, don't be hesitant. We have two experts here, Dr. Prakit or Dr. Christians. They have already presented some valuable information concerning their own domain. Dr. Prakit especially, he has a domain in genetic, in breeding, and Dr. Christians, you are one of researchers in NIFOR, in NIFOR, sorry, especially in development and execution of multidisciplinary research. So don't be hesitant to ask something. We still have some minutes, because this is also give me surprise between Dr. Prakit and Dr. Christian. You presented exactly in the same time. I mean, you spend 17 minutes, both of you. Maybe before you have already discussed. No, it's just kidding. Okay, so I would like to invite again, because we still have the time. From me, in waiting, so maybe some questions will come after. So I just want to know, because especially for Dr. Christians, I'm interested with your expressions. You use the expressions weapon against the poverty, in your slide. Can you hear me? Hello?
He is mute.
Yeah, you still mute, Dr. Christians. Yeah,
can you speak up louder?
Okay, I will repeat again. I'm interested with your expressions in your slides. You wrote this kind of species, the weapon against the poverty. So to show to the audience, to show to us the importance of this kind of species. So my question is, how can you compare the importance of this species with your staple food? You can answer in terms of economic development or the economic status of this species in your country.
Can you hear me, sir?
Pardon? Sorry.
Thank you for your. Can you hear me?
Yes, yes.
Okay. If I get you correctly, you're trying to find out the comparison between the species of the shade tree in terms of economic importance. Is that correct?
Pardon? Sorry.
I said, if I got your question correctly, you're asking for maybe a comparison between the economic importance of the species I have talked about.
Exactly.
Okay, fine. Now, like I try to explain, this commodity, this shade tree, is a commodity that was handed over to the institute, for which there's still ongoing research from the breeders and those that are into tissue culture work. The proper species for this shade tree has not been properly classified. Now, in the beginning of the presentation, I presented to you there's no proper data for now, because what is being monitored is what is gotten from the wild. Now, there's ongoing research work trying to see how this commodity can be domesticated. So, if I make telling you the economic importance of what is available now, this report I give does not identify a particular species, because the information I have given is from what has been gotten from the wild. If there's any new planting because of the effort the Nigerian Institute of Oil Palm Research has made through grafting to reduce, the shade tree, as a commodity is a tree that has a very long gestation period. In my country, in Nigeria, before this commodity was handed over to improve on its growth and development through research, it was known that when you plant it, it will stay for almost 50 years before you start getting the fruits. Now, through grafting in the Nigerian oil palm research, for those seedlings that have been mass produced and distributed to farmers, it is in the next 4 to 5 years that there has to be data. And we'll now start talking about variety. But for now, I cannot tell you that this specific variety is better than the others. But they have identified through survey that this tree is this tree is that. And they have collected what I have presented in those variety that is better. It's what is available in the wild. And they have been brought back to the institute to start working on. I hope I've made myself clear on this, sir.
Yeah, this is challenging for us. So it means for your country especially. Yeah. Okay. Thanks much, and thanks a lot for your answer. Even your answer very detailed. Yeah. Okay. And then I will invite again. Normally we can finish this sessions because we have spent 20 minutes discussions, but we still have more minutes. So I'm going to invite again participants who would like to ask something. Please don't be hesitate to ask directly in the Zoom meeting, or you can just write in the chat Zoom, because I'm not sure. Sometimes this kind of what I will say important time, yeah, for our experts, because Dr. Prakit have many activities, Dr. Christian have many activities. So this is very important time to ask some informations directly from the experts, yeah, from Thailand and Nigeria.
If Dr. Afif,
yes, please.
If there is no question, so I want to discuss in this room about the lab in Indonesia. Do you use this?
Yeah, I will invite some colleagues maybe as for, yes, of course we have it, but maybe several colleagues will explain some informations concerning this kind of species, because I'm personally I didn't do this kind of research, yeah, but I'm very appreciate with your activities in this commodities, yeah, very good, I think. So one of my colleagues maybe would like to discuss, would like to give some informations, like Dr. Prakit Somta would like to discuss in these forums. Yeah, I'm inviting everyone here. Yeah, my colleagues from Brawijaya University.
Me, Pak.
Prof. Kuswanto, maybe, please.
Yes.
Yeah, please, Prof.
Thank you very much.
Yeah.
Yes. Prakit,
yeah,
do you remember? Yeah. Okay. Okay. Actually, many lab in Indonesia. I think this meeting joined Ibu Mrs. Siti Fatimah from University of Trunojoyo in Madura. She said many accession of lab in Madura.
Ah, I see.
Yes, especially in Java, East Java and West Java, many accession of lab.
So you use the young pot and the young seeds.
Yes, young pot, young seed for vegetable. Yes.
Yeah, I will add some information from Kuswanto because like maybe in like yours, yeah, like in your country, normally species or commodities develop more and we use more due to the cultural perspective. Like Prof. Kuswanto said, in one of island in Java, in Madura Islands, there is the culture of the people here consume much this kind of commodities instead of another island. Even instead of another city in East Java. Yeah, I thought you have already been here some years ago.
Yes.
Yeah, in Java. So this is also I think similar with your country. Like in general, in general the species can be cultivated more because of the people interest in the consumption. And one of the islands that often use this kind of species that we call Madura. Have you come there?
Yeah, I've been there.
Ah, okay.
Yes.
So, with Prof. Kuswanto.
Yes.
Yes.
3 years ago, I invite Prakit to Madura.
So Prof. Kus, have you introduced Madurese language to Dr. Prakit? No,
I don't know Madura language.
Even Prof. Kuswanto, you cannot speak Madurese. Okay, I can speak Madurese because my ancient from there. I also like much the lab commodities, yeah, to be consumed. Okay. My ancient from the Madura Island, Dr. Prakit. So I can speak well. Okay, some additional information want to be discussed from you,
Me,
Dr. Prakit?
Pak Afif Darmawan.
Yeah. Pak, yes, please, Pak Dar, you want to ask?
Yeah,
you
hello,
want to?
Prakit.
Yeah,
Yeah, nice to meet you again.
Yeah.
Yeah, maybe I just want to give some additional information that we have one kind of a lab in Indonesia called coral. Yeah, but nowadays there is one kind of coral. Yeah, become booming in Indonesia. We call it coral pedang, but the name of species is not Dolichos laplap. The name of species is Canavalia gladiata. It's a little bit economically high, have a high price in market, and I think this species is prospective for next or future food source in Indonesia. It's just addition information for you.
Okay, thank you. This is good information.
Yeah, thank you, Pak Darmawan, for your additional informations. I give possibility for my colleague here if you want to add some information, because Dr. Prakit asked to be discussed. Yeah? Even for the audience from another university or the other participants, if you want to add some informations, not always to ask something. Yeah, you can give some opinion, you can give some informations that would like to be discussed by Dr. Prakit. Isn't it, Dr. Prakit? Right? Okay, so before finishing these discussions, maybe I will give again the time for Dr. Christian or Dr. Prakit to give some something like closing statement, yeah, concerning your topic that you have present this morning. Starting from Dr. Christian, please.
Thank you once again for giving me the opportunity to make this presentation in this conference. And I've learned a lot by listening. I want to also apologize for the initial technical fault from this angle. The Nigerian economy for now, there's high rise of general price in food commodity. There's a need for the country to diversify the economy. And in diversifying the Nigerian economy, we have to move away from petroleum. And if we're moving away from petroleum, that has been the single source of our revenue for funding our national budget. Every person in Nigeria is looking at diversification to come from the agricultural sector. And the share tree industry, if looked into and properly harnessed, will contribute to the income of this country by increasing its GDP. There is no doubt that research and development into this commodity is still new. Some of the interventions and what we do to promote the growth of this country will only lead if experts collaborate with, I speak for myself, talking from NIFO. Through collaboration, we will see what other countries have done to get a success story. Because for now, there's a lot of unemployment going on in the country. And if people there's awareness to show that these are the investment opportunities that can come from the value chain of the share industry. There is no doubt that poverty, and other vices that the economy is trying to solve to increase productivity per hectare and to ensure food security can be gotten from harnessing the potential of the shea tree industry. Thank you very much for giving me the opportunity to make this presentation.
Okay, thank you a lot, Dr. Christian. It was very remarkable closing statement. Yeah. So you said a little bit concerning a technical problem. It was no problem. In the contrary, we have to say thanks to you because because our condition in the pandemic situation, so we have to do the Zoom meeting like this. Okay, thank you very much again, and I give to you, Dr. Prakit. Please give some statement, yeah, to close this discussion. Please, time is yours.
Okay. When we mention about the underutilized crops, we always talking about their potential for the use and for the economy. But actually, it's really difficult to make the people or or even the farmer or especially the government to realize the true potentials of the underutilized crops. So I think as a researcher, we must try to convince or give the public the information about the true potential of the underutilized crops. Especially in Thailand, in case of Thailand, the government they don't realize the potential of the underutilized crops, so they overlook the opportunity to generate the income or the products from the underutilized species. So I think this is really important. Okay, that is all for my comment.
Okay, thanks much, Dr. Prakit. I think it was very good message from you. Yeah, you give a message for us, or maybe for our country, and also as well as for your country. So that's why I said this is a special message from you. Okay, before we finish, please give warm applause for our expert today. Thanks very much for Dr. Prakit and Dr. Christian. Sorry, and then I have to show to you. This is only symbolics. We are sorry for this symbolic in Zoom meeting. Yeah, this is appreciation from the committee. Yeah, from Brawijaya University, especially from the UCRC. Very especially from the committee, official committee. So thanks to you very much for your time, for your availability, and of course for your valuable information concerning your topic. Yeah, and then for Dr. Christian also. Yeah, please receive first the special soft appreciation certificate. Yeah, from us. This is special gift for the experts, and then as a representative of us to give appreciation and say big thanks to the experts. So I'm as moderator in this last plenary sessions. Once again, I would like to thanks very much to the two experts, who have already explained many things and essential things concerning their topic. And then thank you very much also for all participants who have already focused in these sessions, and especially also for participants who ask something. If you want to ask further, I think you can ask directly to them, maybe by email, for example. Yeah. So the last but not least, I apologize for some mistake maybe. So I give back to the master of ceremony. Thank you very much, and wassalamualaikum warahmatullahi wabarakatuh. Please.
Thank you.
Yeah.
Yes, thank you, Dr. Rafi, for leading a really amazing discussion and also presentations. I would like to say thank you to all the invited speakers for the second sessions and also for all the speakers since day one. So it's really nice and amazing presentation and also discussions. So this insightful and fruitful sessions, we do hope that it will help to embrace a new knowledge, perspective, ideas, breakthrough related to underutilized crops globally and even in Indonesia. And then let's jump to the next agenda that we are waiting for, the announcements of the 3 best speakers for this international conference. So we will show you the video of the best speakers, and then I need to help from Mr. Adit to show the video. So the best speakers go to. Okay, thank you, Mr. Adi, for congratulations to our 3 best presentation for today. And then for the appreciations, we will hand over the certificate. So this is a certificate for the best speakers for today. Mr. Adi. Okay. Okay, so congratulations once again to Mrs. Elina, Mrs. Maya, and also Mrs. or Mr. Kent for winning the best presenter for today. We will hand over the certificate directly to your email address after this. Okay, so maybe we can have a speech from the representative of the best presentation for this international conference. Ibu Maya, may we have your time to give a speech?
Okay, thank you, Vivian. Thank you very much to all the committee. I think I learned a lot from this event, and for the research. This is interesting research. So I learned a lot and have a good experience, and maybe I can improve my research in the future. Thank you very much for all the committee. Thank you.
Thank you very much, Ibu Maya, for giving us a speech for becoming the best presenter for this international conference. Okay, finally we at the end of the international conference for the 2 days, and then before we close the conference, we will have the closing remarks from the chairman of the committee, Dr. Darmawan Saptadi, SPMP. For Mr. Darmawan, time is yours.
Okay, thank you, Mr. of Ceremony. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Dear all participants, we have reached the end of the event and we'll close today's event very soon. Let me express my gratitude to all invited speakers who have shared various information from very diverse fields. We also thank all the participants who have faithfully participated in this event until the end. We are all hope that what we have done will provide the greatest benefit for the development of science and humanity. And as the chairman of this international conference on underutilized crops committee, I apologize if there are inconveniences and mistakes in organizing this event. And finally, by saying Alhamdulillah Rabbil Alamin, we are officially closing this event. Thank you very much and see you next time. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Darmawan, for the closing remarks. So, finishing the closing remark means that we are at the end of our international conference. So, thank you so much once again to all the participants, all the invited speakers who joining since the first day until the second day. And then I just want to remind you to don't forget to fill the attendance list because it will relate to the certificate that will hand over to your email address. Okay, so thank you so much once again for your kind attention. See you again in the next conference. Stay healthy, stay happy, and have a nice day, everyone. Wassalamualaikum warahmatullahi wabarakatuh. Good morning.
Thank you. Thank you.
Thank you.
Thank you very much for.
Thank you very much.
Underutilized Crop Research Center. UCRC, Faculty of Agriculture, University of Brawijaya is one of the lecture and student research activity units that focus on underutilized or neglected plant species.
Yeah, thank you.
Some level within the local, national, or even international community, the potential to contribute further to the food source mix than they currently do. Steps are needed to promote UC through various media in order to provide an optimal role. Communities and local wisdom help to the preservation and